Now, when we consider the ways in which different organisms carry out gas exchange, a huge part of determining their type of gas exchange is their surface area to volume ratio. So how do we calculate this value? Well, we do surface area divided by volume. By definition, the larger the organism, the smaller the surface area to volume ratio. So something like a polar bear has a small surface area to volume ratio. A mouse has a much larger surface area to volume ratio. Make sure you can do simple calculations to find the surface area to volume ratio of something like a cube. So surface area, we're going to do length times width times the number of sides of the cube. For volume, we just do length times width times height. Mammals are able to maintain a constant body temperature, but a mouse has a much higher metabolic rate compared with us, humans, with much smaller surface area to volume ratios. The reason why the mouse has such a high metabolic rate is, as I've kind of just said, it has a very large surface area to volume ratio. This means that heat loss occurs very quickly, and therefore it needs a faster rate of metabolism and therefore respiration in order to maintain this constant body temperature. It's because respiration releases heat. That heat is needed to maintain their body temperature. You might have heard of amoeba. They're a type of single-celled protoctist. Why don't they require specialized gas exchange structures? That's because being single-celled, they have extremely large surface area to volume ratios, and therefore diffusion is able to provide all the oxygen they require for their respiration, because diffusion occurs super fast over such a large surface area. Simple diffusion is not sufficient for much larger organisms. That's because they have much smaller surface area to volume ratios. It means that their volume increases at a faster rate than their surface area, and therefore simple diffusion is too slow in order to reach the middle of the organism, which also requires oxygen as well as other substances. Some organisms have evolved in order to maximize the efficiency of exchange. A good example is the flattened shape of many leaves of plants. This means that most cells are located towards the surface, meaning that diffusion can occur nice and quickly. Some organisms have evolved specialized gas exchange surfaces. Humans, for example, have alveoli present in their lungs. Fish have evolved gills. So really, to explain the features of an efficient gas exchange surface, as I've already said, we want a large surface area. This means more surface is provided for diffusion to take place. We want to have a thin surface, so that provides a short diffusion distance, meaning diffusion occurs more quickly. We want a steep concentration gradient, so that means a really large difference between the areas of high concentration versus the areas of low concentration. That will increase the rate of diffusion. The dense capillary network around alveoli is a really good example in humans of somewhere with a steep concentration gradient. Those capillaries quickly remove oxygen and take it quickly to the pulmonary vein, which, remember, is going to enter the heart. Another feature of an efficient gas exchange surface is good ventilation. This also helps to maintain a steep concentration gradient. For example, fish gills maintain a steady flow of water over their gas exchange surfaces, their gills. Uranema marinum is a single-cell eukaryotic organism. The diagram below is a photograph of U. marinum taken through an optical microscope. In large cells of U. marinum, most mitochondria are found close to the cell surface membrane. In smaller cells, the mitochondria are distributed evenly throughout the cytoplasm, so that's going to be due to the large difference in surface area to volume ratios. In larger cells, remember, they'll have a small surface area to volume ratio, whereas in the smaller cells, they'll have a large surface area to volume ratio. And so the mitochondria use oxygen during aerobic respiration. Use this information and your knowledge of surface area to volume ratios to suggest an explanation for the position of mitochondria in large eumarinum cells. So first up, I'm going to say large eumarinum cells have smaller surface area to volume ratios. The knock-on effect of that is that it takes longer for oxygen to diffuse to the mitochondria. So that's why we need to reduce that diffusion distance as much as possible so that the mitochondria are located close to cell surface membrane. But I've said enough there for 2 marks. Explain the advantage for larger animals of having a specialised system that facilitates oxygen uptake. Again, it's the same point every time. Larger animals have small surface area to volume ratios, specialised gas exchange systems overcome the issues of this long diffusion pathway. Remember, the larger the surface area to volume ratio, the shorter the diffusion pathway, so that's why we're arguing the need for the specialised gas exchange system. Figure 1 shows two models of oxygen uptake found in animals. So in A, we've got oxygen uptake through a system developed to the outside of the body, such as fish gills, versus oxygen uptake through system developed to the inside of the body, such as human lungs. This is looking very like an alveolus. Suggest how the environmental conditions have resulted in adaptations of systems using Model A and Model B. Well, we've got to think about where these two organisms live. Fish live in water, and water is denser than air, meaning that water supports the presence of gills in fish, whereas mammals live in air. You could have also pointed out that water contains a lower oxygen concentration compared with air, and therefore having this system where the gas exchange surface is on the outside kind of counteracts the issue with the lower oxygen concentration in water, because it provides a larger surface area for gas exchange to take place over. That means, therefore, the table below shows features of two mammals. Bats are flying mammals, shrews are ground-living mammals. Calculate how many times the lung volume per unit of body mass of the bat is greater than that of the shrew. Give your answer to an appropriate number of significant figures. So we need our answer per unit of body mass. So although we're looking for volume, we need to divide that volume by the mean body mass in order to get our answer per unit of body mass. So we do 12.48 divided by 0.096, and we do 0.72 divided by 0.024. And then to work out how many times greater the lung volume of the bat is compared to the shrew, we take that bat figure, divide it by the shrew figure to get 4.3 recurring. We need an appropriate number of significant figures, so look at the fewest number of significant figures provided in the data. It's 2. So that means that our final answer has to also be to 2 sig fig. Give one suggestion to explain the difference. The reason for this difference is that it provides more oxygen for respiration. We're going to take insects now and look at their specialised gas exchange surfaces. So let's first of all look at why insects need a totally different gas exchange system compared with mammals. First up, I keep mentioning it, they have a very large surface area to volume ratio. Secondly, if you think about their bodies, they have what's known as a tough exoskeleton, so it's that kind of hard shell that we think of. The presence of this exoskeleton means that gas exchange can't take place directly through it. And lastly, they have an equivalent of blood, but they don't contain blood pigments which can transport oxygen, so they have no haemoglobin. So if we look more closely at the structure of the gas exchange system in an insect, it consists of a network of tubes with many branches, and where those branches reach the kind of exoskeleton of the insect, you'll find small sphincters. These are holes which can open and close, and they have a special name in insects called spiracles. So these spiracles are located in the thorax and the abdomen of the insects. They allow air to enter and leave the tracheal system. We'll look at trachea soon. And actually, when those spiracles are open, it allows water to leave. So what are tracheae? They're a network of air-filled pipes that run along the insect's body. As I've already said, they have spiracles at the end, which can open and close, and a network of chitin helps to maintain the tracheae structure. Tracheoles is the name given to the narrow branches of the tracheae. Most of the gas exchange in insects takes place in these tracheoles. Present in the tracheae and tracheoles is tracheal fluid. The purpose of this fluid is to limit the penetration of air for diffusion. Now, the tracheae and the tracheoles are incredibly well designed so that sometimes they're filled with fluid and sometimes they can be filled with air. And whether they're filled with air or fluid depends on the oxygen requirements of the insect. So when the oxygen demand is high, lactic acid causes the tracheal fluid to leave the tracheoles by osmosis. By doing so, it means that more surface area is exposed for gas exchange. And the knock-on effect is it draws more air in to the tracheoles for gas exchange. How does the oxygen reach the cells of the insect? Well, the tracheals run past each other, and therefore the oxygen is able to diffuse from the tracheals into the insect cells. So how is gas exchange controlled in insects? It's through the opening and closure of the spiracles. You can almost think of spiracles like the stomata in leaves. So if the oxygen demand is high by the insects, the spiracles are open, such as the case with the stomata. If they need a lot of carbon dioxide for photosynthesis, the stomata are open. We know that water can leave both the stomata and the spiracles, and in order to minimize the water loss, the insects can close their spiracles at times when their need for oxygen is low. I know that the specification is super detailed here, but they do want you to know how larger insects cope with their increased demands for gas exchange. So one thing the large insects can do is carry out mechanical ventilation. This is where air is actively pumped into the tracheal system through the muscular contraction of their thorax and abdomen. By doing this, it changes the pressure in the trachea, forcing air in and out of the insect. A second adaptation is the presence of collapsible air sacs in the tracheal system. These can act as air reservoirs, and whether they're inflated or deflated depends on movements of both the thorax and the abdomen. So what are the relative advantages and disadvantages of the tracheal system? Well, first of all, the main advantage is the direct delivery of oxygen to insect cells and the direct removal of carbon dioxide. There are quite a few disadvantages though. Obviously, the presence of a tracheal system limits the size of the organism, which is why even when we think insects are pretty scary, pretty big, like hornets, they never become massive. An insect will never become the size of us or an elephant. Coupled with this, the tracheals can only successfully deliver enough oxygen if the diffusion distance is short and if there's a pumping mechanism by the abdomen in order to keep the air moving. So again, that means we can only have a short distance, therefore limiting the size of the insect further. I'm sure you can already see some similarities, however, between the insect's gas exchange system and mammals' gas exchange system. You've got the presence of thin walls, large surface area, short diffusion distance. Several of the linings are moist to enable gases to dissolve, and we have a steep concentration gradient maintained in both by ventilation. That in mammals, oxygen is transported to respiring tissues via a circulatory system consisting of the blood, the heart, connecting vessels. In mammals, the alveoli are the gas exchange surface in mammals. In insects, it's the junction between tracheals and respiring cells. Figure 1 shows the stages of development of an insect called a damselfly. The adult damselfly uses a tracheal system for gas exchange. Explain 3 ways in which the insect's tracheal system is adapted for efficient gas exchange. Let's start by saying that the tracheals have thin walls. We have to explain our answer, so say why that's of benefit. So the diffusion distance is short. Let's say that the tracheal fluid can move in and out of the tracheals to provide more oxygen in vigorous activity. Tracheals are highly branched to provide a large surface area to volume ratio for gas exchange. The damsel larva is a carnivore that actively hunts prey. It has gills to obtain oxygen from water. Some other species of insect have larvae that are of a similar size and shape to the damsel larvae and also live in water. These larvae do not actively hunt prey and do not have gills. Explain how the presence of gills adapts the damselfly to its way of life. The fact that the larvae is a carnivore means that it's actively having to hunt its prey, as it says here. So it's going to need to release a lot of energy in order to do that. So we're going to say that the damselfly larvae have a higher respiratory rate to release the energy needed to hunt prey. A scientist measured the size of each gill lamella of the gills of 40 damsel larvae. His results are shown in the table. Calculate the mean surface area of one side of one gill lamella. Assume that a gill lamella is rectangular. Give your answer to an appropriate number of significant figures. Include the percentage error uncertainty of a surface area in your answer. Show your working. So I'm going to start by finding the percentage uncertainty. So we take the uncertainty, which is 0.19, and then divide it by the reading, 1.61. There's the first percentage uncertainty. Let's do the same with the width. Take the uncertainty, divide it by the reading. Our total percentage uncertainty now is those two numbers added up. Now I'm going to find the mean surface area of one side. So I'll take 1.61 multiplied by 6.12. So that's the two widths multiplied together. And I've already worked out the percentage uncertainty. The respiratory system of a bony fish. We're going to start with the fish's mouth, which we're going to call the buccal cavity. What's special about the buccal cavity is that its floor can raise or lower, and that's going to be super important in helping to draw water into the fish's mouth. And later on, it's going to help draw that water over the fish's gills. The operculum is the name given to a flap which covers the fish's gills. The gills themselves are made up of gill filaments. These gill filaments occur in stacks known as gill plates, and the gill filaments are separated by flowing water. And the arrangement of these gill filaments helps to maximize the surface area. One thing they do like to regularly ask you in the exam is about the fact that the gill filaments overlap slightly, and that's in order to slow the movement of the water to increase how much gas exchange can take place. The gill lamellae are organs of gas exchange. They're located on the gill filaments, and as you'd expect, they have a large surface area and a rich blood supply. They're also very thin, and this provides a short diffusion pathway. So we know that water needs to enter the fish's mouth before it can then wash over its gills so that gas exchange can take place. But what processes take place in order to allow this? The first thing that happens, and this is the equivalent of a breath in that we take as humans, is that the fish opens its mouth and lowers the floor of its buccal cavity. By doing so, it increases the volume within the fish's mouth, therefore decreasing the pressure. So you'll see similarities when we consider gas exchange in humans, all to do with pressure and volume within the thorax. But as a result of the increased volume in the buccal cavity, the lowered pressure, water is moved into the buccal cavity. But how does that water then move from the buccal cavity so that it then washes over the gills of the fish? Well, this time the floor of the buccal cavity is raised. This decreases the volume, increasing the pressure, meaning that water is forced out of the mouth and over the gills. And lastly, out of the operculum. Fish are incredibly well adapted in that they have a countercurrent flow system, and this helps them to maximize the amount of gas exchange that takes place. And what that really means is that the water flows in the opposite direction to the blood flow. This maintains a constant concentration gradient along the length of the gill and ensures that water is always flowing next to blood with a lower concentration of oxygen. So to summarise, how is the structure of the bony fish adapted to maximise the amount of gas exchange that can take place? We've already said that gills have large stacks of gill filaments that contain gill lamellae. These gill lamellae have an incredibly large surface area. They're thin to provide a short diffusion distance, and they have a good blood supply. A constant flow of water is maintained over the gills, and this maximises the concentration gradient of the respiratory gases. The tips of the gill filaments overlap in order to slow down the flow of the water to increase the amount of gas exchange that occurs due to the increased resistance to the water flow produced by the overlapping gill filaments. And the countercurrent exchange system makes sure that blood is always flowing next to water that contains a higher concentration of oxygen. So looking more closely, here we can see obviously the fish. Here's its mouth, its buccal cavity, for increasing and decreasing the volume within the fish's mouth, which will therefore help draw water into the fish and then enable it to flow over the gills. The gills consist of a stack of gill filaments. If we look more closely at these filaments, they contain gill lamellae, which increase the surface area, allowing more gas exchange to occur. Remember, we said that water flows in one direction compared with blood flow, and we're going to look at that in more detail now. So here we're comparing parallel flow system versus the bony fish's countercurrent flow system. So this first example of the parallel flow system, we can see that the direction of the blood flow and the direction of the water flow are the same. So we'll take each of these circumstances in turn so that we can understand why the countercurrent flow system ensures that much more gas exchange can occur for that bony fish. So in this situation, we know that water entering the fish will have a high concentration of oxygen, as we see here. The blood flow through the gills, because that oxygen has been used by the fish in respiration, it's going to be a low concentration of oxygen. So initially, we can see that there'll be a lot of diffusion of oxygen from that water to the blood. But as that blood therefore becomes more oxygenated, we know that the water will contain less oxygen. So you reach an equilibrium really soon, as we see here. And if you do chemistry, you'll recognise the chemical equilibria graph we see here. And at that point, where the concentrations of oxygen are the same in the blood and the water, then we know that no further diffusion occurs. So actually, it's really only at the start of this process that gas exchange can take place. Whereas if we take a countercurrent flow system, what's so clever about this is we know that the water and the blood are flowing in opposite directions. So down here, because this is downstream of the water entering, we know that there'll be less oxygen here. But because this blood is coming from the tissues of the fish, we know that that oxygen will have been used up in the fish's respiration. So we know that the blood here will also contain a very low concentration of oxygen, and in fact it's lower compared within the water, because after all, oxygen is always dissolving in water, so diffusion will occur. Then as the blood has more oxygen in it, it reaches water which contains more oxygen. Again, the water always contains more oxygen compared with the blood, so diffusion occurs here. And then even up here, when the fish's blood contains lots of oxygen, well, look, this is where the water contains the most amount of oxygen. It hasn't been depleted yet. So again, diffusion takes place here. So throughout the whole process, the capillary near the lamellae, diffusion is occurring throughout. And this graph really shows very nicely how the water always has a higher concentration of oxygen compared with the blood. And it doesn't matter what point along the gill plate we're looking at, that remains the same. So diffusion continued to occur. Whereas here, yes, initially diffusion occurs really quickly due to the steep concentration gradient between the water and the blood. But as we move along that gill plate, we reach an equilibrium point where diffusion will no longer take place. And the last thing I want to point out is, why is efficient gas exchange so important by the fish? It's due to the properties of water. Water is denser and more viscous than air, and water has a much lower oxygen content than air. Below is an image of a fish gill taken using a scanning electron microscope. Identify structures labelled F and G. So F is our gill filament, whereas G are our lamellae. Describe and explain the advantage of the countercurrent principle in gas exchange across a fish gill. So we're going to start by saying what it is. So we're going to say that water and blood flow in opposite directions. This means that the oxygen concentration is always higher in water, meaning that diffusion of oxygen can occur along the whole length of the gill. Figure 2 shows changes in concentration of oxygen in two gas exchange systems. Just make sure you look at the graph we have. On our y-axis, the concentration of oxygen compared with time. Here's our human gas exchange system. This is our fish gas exchange system. You'll have seen a similar graph to this when I showed you the countercurrent airflow system of the fish. You can see nicely here the parallel flow system versus the countercurrent flow system. So I stands for the concentration of oxygen in the air entering the gas exchange system in the human. E stands for the concentration of oxygen in the air leaving the gas exchange system. A stands for the arterial blood entering the gas exchange system's oxygen concentration. V is the venous blood leaving the gas exchange system. Obviously, we're going to swap air to water for the fish. A student studied figure 2 and concluded that the fish gas exchange system is more efficient than the human gas exchange system. Use figure 2 to justify this conclusion. So we're using figure 2 to make statements which agree with the fact that the fish gas exchange system is more efficient. So in terms of justifying this conclusion, have a look at the numbers. You can see that in fish, the blood leaving the gas exchange system has a higher oxygen concentration than the water leaving, whereas the opposite is true for humans. The air leaving has a higher concentration than the venous blood. Explain how the countercurrent principle allows efficient oxygen uptake in the fish gas exchange system. They love asking this. Remember, we're saying that blood and water flow in opposite directions. Meaning that water always has a higher concentration of oxygen. So diffusion occurs along the whole gill plate. Gas exchange in plants. To begin with, we need to know the detailed structure of a leaf. Remember, the leaf is the main organ of photosynthesis. This is where carbon dioxide and water is used to make glucose and oxygen. So we're going to go through the various layers of the leaf, starting with the topmost layer, the waxy cuticle. This helps prevent loss of water by transpiration. Then we have the upper epidermis, which is transparent to allow light to enter the leaf. The palisade mesophyll contains many chloroplasts needed for photosynthesis, and the cells here are arranged vertically and they're tightly packed in order to absorb as much light as possible. Next up, we have the spongy mesophyll. This contains air spaces which allow gases to diffuse. The stomata are small pores located to the bottom surface of the leaf. These allow carbon dioxide to enter and oxygen and water to leave. The stomata's opening and closure is controlled by guard cells. So how do plants carry out gas exchange? Well, it's the stomata and the guard cells in the leaf which are incredibly important. So carbon dioxide and oxygen are able to enter and leave the leaf via the stomata by diffusion, and it's the process of photosynthesis which maintains the gas concentration gradients within the leaf. Think about the sorts of processes that are more likely to take place during the day and night. So obviously at night time, no photosynthesis can take place due to the lack of light, so therefore the guard cells close the stomata to prevent excess loss of water, which could lead to the wilting of the plant. During the day, when there's lots of light, lots of photosynthesis will be occurring. The guard cells open the stomata to allow as much carbon dioxide into the leaf as possible. The oxygen and carbon dioxide diffuse through the air spaces to the spongy mesophyll, to the palisade mesophyll where they'll be used, and carbon dioxide dissolves in moisture present in the mesophyll cell walls. So how are leaves adapted for gas exchange? Well, first of all, they have lots of stomata, and no cell is actually very far from a stomata, meaning that there's a short diffusion pathway. There are many interconnecting cells, which increases the exposure of mesophyll cells, and the large surface area of mesophyll cells increases the rate of diffusion. So why do plants grown in soil that contains very little water grow so slowly? That's because the stomata are often closed in order to prevent excess water loss. After all, we know there won't be much water coming in through the roots. With the stomata closed, carbon dioxide can't enter the leaf, and therefore very little photosynthesis can take place. Little glucose is made, little growth occurs. Now, xerophytes are plants which are particularly well adapted to life in very dry conditions, such as marram, grass and cactus. Various adaptations of xerophytes to live in very dry, arid conditions include the following. They have very thick waxy cuticles to prevent water loss by evaporation, and it actually also increases the diffusion distance. They often have spines instead of leaves. This means there's a smaller surface area, so less evaporation can take place. And the fact that you have spines means that there'll be fewer stomata in existence, meaning less transpiration occurs. Many xerophytes have hairs on their leaf. The hairs reduce airflow and therefore help to trap water vapour close to the leaf. That in effect creates humid conditions, which remember reduces the rate of transpiration, as there's a less steep concentration gradient between the outside atmosphere and inside of the leaf. Sometimes you find that stomata are in pits. Again, this will increase the humidity, reducing that water potential gradient. Many xerophytes have long, shallow root systems. That's in order to obtain that water as quickly as possible if it ever rains. And succulents, I actually own quite a few succulents myself. These have specialised tissue which is able to store large amounts of water in both the stems and the roots. The water potential of leaf cells is affected by the water content of soil. Scientists grew sunflower plants. They supplied different plants with different volumes of water. After 2 days, they determined the water potential in the leaf cells by using an instrument that gave a voltage reading. The scientists generated a calibration curve to convert the voltage readings to water potential. Figure 1 shows their calibration curve. Sunflowers are not xerophytic plants, so they're not adapted to live in dry conditions. The scientists repeated the experiment with xerophytic plants. Suggest and explain one way the leaf growth of xerophytic plants would be different from the leaf growth of flowers in figure 2. I think we would all agree that leaf growth would be slower in xerophytes, and the reason for that is because they have fewer stomata, which means they can absorb less CO2 and therefore less photosynthesis takes place. Use your knowledge of gas exchange in leaves to explain why plants grown in soil with very little water grow only slowly. I already said this in my video earlier. So little access to water means that stomata close to prevent wilting. When stomata close, less CO2 is absorbed, meaning less photosynthesis takes place. Gas exchange in mammals. This is the process whereby oxygen enters the capillaries from the alveoli and carbon dioxide leaves the blood. Features of a mammalian gas exchange system include the following. So we've got trachea, bronchi, bronchioles and alveoli. The role of our trachea, our windpipe, is to carry warm, moist air from our nose to our chest. To keep the trachea open, it's supported by C-shaped rings of cartilage. These keep our airways open but also allow food to pass down our esophagus, which remember is our food pipe located very close to the trachea. Trachea contains smooth muscle which can contract in order to narrow the lumen of the trachea, whereas elastic fibres do the opposite and they allow the lumen to dilate. Other features of the trachea include the presence of goblet cells and ciliated epithelial cells. The role of the goblet cells is to secrete mucus which traps pathogens and dust. The ciliated epithelial cells have cilia, small hairs, which help to waft that mucus laden with bacteria up the trachea to the mouth, where it then gets swallowed and destroyed by hydrochloric acid in the stomach. When the trachea branches, we enter the left and right bronchus. These are supported by smaller C-shaped rings of cartilage. Smooth muscles again help to narrow the lumen. Elastic fibres help to dilate the lumen. Further on, we have even more branching. These branches are known as bronchioles. They lead from the bronchi to the alveoli. They're made from smooth muscle and elastic fibres, which again, their role is to constrict or dilate the lumen. And then lastly, the alveoli are grape-like structures where gas exchange takes place. They have a very large surface area, and we'll talk lots about their adaptations very shortly. They have elastic fibres which enable the alveoli to stretch and recoil so that it can return to its original shape. So specifically what takes place at the alveoli? Well, we know that oxygen will diffuse from the alveoli to the capillaries. Carbon dioxide will diffuse in the opposite direction. The oxygen that enters the capillaries binds to the haemoglobin and red blood cells. The volume of the alveoli increase when you take a breath in and inhalation. And it's important that concentration gradients are maintained so that oxygen diffuses quickly into the blood and CO2 diffuses quickly out. So the adaptations of the alveoli to maximise gas exchange include a very large surface area to volume ratio. They have thin walls to provide a short diffusion distance. They're moist, they're lined with something known as lung surfactant. This allows gases to dissolve and keeps the alveoli inflated. They have a good blood supply provided by plenty of blood capillaries. This helps maintain that steep concentration gradient, and they also have good ventilation, and it's through the process of breathing that that concentration gradient is further maintained. Now, fibrosis of the alveoli will restrict our ability to breathe properly. It will reduce the amount of gas exchange that occurs, and that's because the scar tissue that forms means that the alveoli lose some of their elasticity, meaning they're less able to recoil during inhalation and exhalation. So the process of ventilation just simply means inhalation and exhalation of air between the lungs and the surrounding atmosphere. The role of ventilation is to maintain the concentration gradients of oxygen and carbon dioxide. It provides a steep concentration gradient whereby the blood capillaries have a low concentration of oxygen compared with the alveoli, which have a high concentration of oxygen. The opposite is true for carbon dioxide. We have a low concentration in the alveoli versus a high concentration in the blood. The process of ventilation is as follows. So we'll start with a breath in and inhalation. We need to consider what happens in terms of volume and pressure within the thorax. So to begin with, the external intercostal muscles contract. This moves the rib cage up and out. The diaphragm contracts, it flattens. Through these two processes, the volume in the thorax increases. Because the volume is much greater, the pressure decreases. You have a pressure gradient whereby the surrounding air has a higher pressure, meaning that air gets forced into the lungs. During a breath out and exhalation, the opposite occurs. The internal intercostal muscles contract, so the ribs move down and in. The diaphragm relaxes and becomes domed in shape. Together, these decrease the volume within the thorax, meaning that the pressure has subsequently increased compared with the outside air, and therefore air is forced out of the lungs. And furthermore, our abdominal muscles can be used to produce a stronger exhalation. One last definition is that of antagonistic muscles. These are pairs of muscles whose action opposes each other. For example, the circular and radial muscles in our iris have opposing effects on the pupil's diameter. Describe and explain one feature of the alveolar epithelium that makes the epithelium wall adapted as a surface for gas exchange. Do not refer to surface area or moisture in your answer. So you want to say that the epithelial wall is thin. That noise, by the way, is tropical India. Sounds very different from my home in Cornwall. This reduces the diffusion distance of carbon dioxide and oxygen. Doctors measure the health of lungs by calculating the FEV1-FVC ratio. FEV1 is the maximum volume of air exhaled in 1 second. FVC is the maximum volume of air exhaled in one breath. The minimum FEV1-FVC ratio of healthy lungs is 0.7 to 1. A man with the lung disease emphysema inflated his lungs fully. He then exhaled as much of this air as quickly as possible in one breath. The figure below shows how the volume of exhaled air changed during this breath. Use the information provided to determine the FEV1-FVC ratio of this man's lungs. Go on to determine how many times greater the minimum ratio of healthy lungs is than his ratio. So start by drawing lines on the graph using the information here where it says that FEV1 is the maximum volume of air exhaled in 1 second. So I've already drawn a line up here from 1 second. I met the graph and now I'm going to read across so I can see that that's 2.2 decimeters cubed. The second definition is that FVC is the maximum volume of air exhaled in one breath. And have a look at the graph. Look, that's the maximum point in the graph. It must be the maximum volume of air exhaled in one breath. Read that carefully. It's 3.6 decimeters cubed. Then we need to work out the ratio, which is worked out by doing FEV1 divided by FVC. So that's 2.2 divided by 3.6. We need it to be a ratio, so that needs to be 0.6 to 1. And so finally, to work out how many times greater the minimum ratio of healthy lungs is than his ratio, use his ratio, which was 0.7 to 1. So just take that 0.7 divided by our number we just calculated, so we know it's 1.15 times greater. Tidal volume is the volume of air inhaled and exhaled during a single breath when a person is resting. The tidal volume in a person with emphysema is reduced compared with the tidal volume in a healthy person. Suggest and explain how a reduced tidal volume affects the exchange of carbon dioxide between the blood and the alveoli. Make sure you have a look at the information provided here. It's asking us about carbon dioxide. So if we have reduced the volume of air being exhaled when you have emphysema, it means that less CO2 will be exhaled. That means that the concentration gradient will be shallower between the blood in the capillaries and the alveoli, meaning that there's less movement of carbon dioxide out of the blood. Describe the pathway taken by an oxygen molecule from an alveolus to the blood. So the oxygen molecule crosses, be really specific here, the alveolar epithelium, which is only 1 cell thick. The oxygen molecule enters the epithelium of the capillary. This is a short diffusion pathway. Explain how one feature of the alveolus allows efficient gas exchange to occur. The alveolar epithelium is 1 cell thick, creating a short diffusion pathway. Carbon monoxide is a poisonous gas that is present in cigarette smoke. This carbon monoxide can be absorbed into the blood where it binds with haemoglobin. Scientists investigated the concentration of carbon monoxide in cars in which people were smoking or not smoking. They measured the concentration with the car windows open and closed. The graph shows the scientists' results as they presented them. A value of plus or minus 2 standard deviations from the mean includes over 95% of the data. So on the y-axis we have the mean concentration of carbon monoxide in the air inside the car. And then we have no smoking versus smoking for 5 minutes and 5 minutes after smoking, as well as the key showing the white bars represent an open window and the grey bars represent a closed window. And here these error bars represent plus or minus 2 standard deviations. So it is obviously important that you know what standard deviation means. It's a measure of how spread out our data is. So the greater the standard deviation, the greater the spread of data. Have a look closely at the error bars here. You can see an overlap here between the 2 error bars. And because those error bars are overlapping, it shows that there's no significant difference between the 2 results. And you'd expect that because look, if we're not smoking, why does it matter if the window's open or closed? That's why there's no significant difference here. If we look here, smoking for 5 minutes, look, there's no overlap between the 2 error bars, meaning that there is a significant difference between the results, so we can draw some conclusions there. And the same is true here, there is no overlap again, so there is a significant difference. In England in October 2015, a law was introduced making it illegal to smoke in a car carrying someone who was under the age of 18. Following the introduction of the law, a politician stated, "It is dangerous to smoke when a child is in the car. High levels of deadly toxins can build up even on short journeys, and children breathe faster than adults, meaning that they inhale more of the deadly toxins. Use the information provided and the data in the graph to evaluate the politician's statements." That's worth 4 marks. As always, because we're evaluating the data, we're going to say arguments for and against, and then comment on the quality of the study. So what about the data here supports the statement that it's dangerous to smoke when a child is in the car? First thing you can see is that there are significantly higher concentrations of carbon monoxide in these 2 sets of data compared with no smoking. And we know that, as I've already said, because there's no overlap of the error bars within 2 standard deviations. So there is significantly more carbon monoxide compared with no smoking with closed windows, and it is significant because there's no overlap in 2 standard deviations. And you can see that these carbon monoxide levels remain high even 5 minutes after smoking. And let's say why that's a problem. Carbon monoxide stops oxygen being transported by haemoglobin, particularly in children. So I'm using the politician's statement as they breathe faster. And then if we look more closely at these error bars, just for the open window, look, there's no significant difference between them because they're overlapping, which means that the data suggests that smoking with the windows open will have no significant effect on the mean concentration of carbon monoxide. Again, that's quite a difficult mark because we're talking a lot about standard deviation. To be honest, you might prefer to just say, well, actually, I don't even know if the numbers relating to parts per million are even deadly, as they've said here, the politician. So arguments against this statement: no idea if 5 parts per million is deadly, and no data was provided on child breathing rates. So, okay, fine, they're breathing faster, but if their tidal volume is less, then it wouldn't make any difference. Digestion. Happily, I think this is one of the more straightforward topics. Let's start by differentiating between physical and chemical digestion. Now, the whole point of digestion is to break down the food that we eat into soluble food particles that can then be absorbed through the wall of our small intestine. So we're just building on the information you learnt at GCSE and IGCSE. Let's have a look at this simple diagram of the digestive system. We're going to start at our mouth. Then we move on to our food pipe, our esophagus. That leads into the stomach. Then we have the small intestine, the large intestine, the rectum and the anus. Our pancreas is an important organ which produces enzymes. Our liver will have important roles to do with bile. Part of the small intestine is known as the ileum, and we will need to add a little more detail here. So starting with the definition of physical digestion, the purpose of physical digestion is to break down large pieces of food into smaller pieces of food. We don't change the structure of the food, but we do increase its surface area to volume ratio. This largely takes place in our mouth through the action of our teeth chewing and biting, as well as our stomach, where muscular contraction helps to break down those food pieces even further. The movement of food down our esophagus and along our gut is known as peristalsis. Here you'll find waves of contraction of both circular and longitudinal muscles which help to move that bolus of food in the appropriate direction. Chemical digestion is all to do with enzymes, and it's about altering the structure of our food. Here enzymes are going to take large insoluble food molecules and they're going to hydrolyze them into small soluble food molecules. And we'll look at specific examples shortly. The first enzyme we'll meet is amylase. This hydrolyzes starch into maltose. Amylase is produced by both the salivary glands and the pancreas, and amylase acts in the mouth and the ileum, which is part of the small intestine. Maltose is a disaccharide. It needs to be hydrolyzed further by the action of maltase enzyme. Maltase hydrolyzes maltose into the monosaccharide glucose. Maltase is produced by the ileum. Maltase is known as a membrane-bound disaccharidase. Think about what those individual words mean. Maltase is known as membrane-bound because it attaches to the cell surface membrane of the ileum, epithelial cells. It's a disaccharidase because it hydrolyzes a disaccharide maltose into a monosaccharide glucose. Sucrase enzyme now, this hydrolyzes glycosidic bonds in the sugar sucrose. By doing so, it produces glucose and fructose, which are both monosaccharides. Moving on to lipase, another digestive enzyme. This hydrolyzes lipids into fatty acids and monoglycerides. Lipase is produced by the pancreas, but it acts in the ileum. What about bile now? Well, at higher level biology, we need to refer to them as bile salts. As you would expect, bile salts emulsify fats or lipids. They create micelles, and we'll talk a lot about micelles very shortly. Bile salts increase the surface area of lipids, making the action of lipase more efficient. And the second role of bile salts is to neutralize hydrochloric acid in the stomach, providing an optimum pH for enzymes released into the ileum. 3 different proteases now. At high level bio, we're going to call them peptidases. First up, exopeptidase. Exo, they're going to act at the end of the polypeptide chain. So they will hydrolyze the peptide bonds holding together the final 2 amino acids, so the individual amino acids will be released, as well as small dipeptides. So that means polypeptides made up of just 2 amino acids. Endopeptidases hydrolyze peptide bonds located in the middle of the polypeptide chain. Endopeptidases are produced by the stomach and the pancreas, and they are released into the stomach where the pH will be low because of the presence of the hydrochloric acid, as well as being released into the ileum. Dipeptidases is the name given to membrane-bound peptidase enzymes located in the ileum. Dipeptidases hydrolyze peptide bonds in dipeptides, producing single amino acids, and then these amino acids are then able to cross the cell membrane. Many herbivores, such as rabbits, will eat diets high in cellulose. It's not possible for us to eat grass, which contains a lot of cellulose, and that's because we don't have the enzymes needed for that cellulose's digestion. It's obviously important that we can break down these large molecules into smaller, more soluble ones so that they can be absorbed through the walls of the villi, and then these small food molecules can be assimilated, build up to make larger, more useful molecules. So how are the various products of digestion absorbed into the blood at the small intestine? First of all, by simple diffusion, so moving from an area of high concentration to low concentration. Facilitated diffusion again occurs from high to low concentration, but will require the use of transport proteins, and amino acids may use facilitated diffusion to enter the blood. Active transport moves from an area of low concentration to high concentration, requiring ATP, such as glucose. Endocytosis involves enclosing our substance in small vesicles, such is the case with fat-soluble vitamins, and co-transport, such as the sodium pump, can be used to move glucose. So how is the lining of the small intestine adapted for the absorption of soluble food molecules? Well, it has lots of infoldings, known as villi, together with microvilli, which massively increase the surface area for absorption. The thin walls of the epithelium allow a short diffusion pathway. A good blood supply rapidly removes the digested food molecules, maintaining a steep concentration gradient for diffusion. A lacteal is present in the villus, which is used for fat absorption. There are many protein pumps and protein channels located in the membrane, which can be used in the active transport and facilitated diffusion of substances. And the presence of many mitochondria provide the ATP needed for active transport. So in the exam, if they expect a more detailed answer as to how amino acids and glucose are absorbed through the wall of the small intestine, you'll need to say the following. So starting with the need for co-transport, which is when sodium ions are actively pumped out of the epithelial cells into the blood. This occurs through the sodium and potassium pump and requires ATP. Because the concentration of sodium ions is greater in the lumen compared with the epithelial cells, sodium ions diffuse into the epithelial cells through a co-transport protein. And because they're moving down their concentration gradient and it involves a co-transport protein, we say that this occurs by facilitated diffusion. As the sodium ions move, they take glucose or an amino acid with them into the epithelial cells, and the amino acids and the glucose, because they're moving against their concentration gradients, this is an example of co-transport, and then the glucose and amino acids then diffuse from the epithelial cell into the blood by diffusion. So why are we saying that it's kind of all three, active transport, co-transport and diffusion? Because the movement of sodium ions in the first instance required ATP, and then the glucose, as it gets transported back with the sodium ions, is moving against its concentration gradient, so that's why it's active transport. But because the sodium ions are moving from an area of high concentration to an area of low concentration, we can say that they're moving by diffusion. Due to the involvement of a co-transport protein, it's facilitated diffusion. So how is the energy required for the active transport of sodium ions released? Well, we need to hydrolyze the ATP, so adenosine triphosphate, to adenosine diphosphate plus an inorganic phosphate, and that energy is therefore released through those bonds breaking. We already mentioned micelles when we looked at bile salts, but what are they? They're small droplets formed from monoglycerides, fatty acids, and bile salts, and they're really important in the absorption of monoglycerides and fatty acids through the wall of the ileum in the small intestine. Because the micelles contain a combination of monoglycerides and fatty acids and bile salts, what this does is it makes the fatty acids more soluble in water, and therefore helps to bring the monoglycerides and fatty acids to the lining of the ileum, creating a higher concentration of monoglycerides and fatty acids at that lining of the ileum, so that they are then able to be absorbed by diffusion. Now, I've already mentioned the lacteals as being important in fat absorption. In summary, the role of the lacteals is to transport triglycerides away from the ileum into the lymph. But how do the monoglycerides and fatty acids enter the lacteal? Inside the epithelial cells of the lining of the ileum is smooth endoplasmic reticulum. The monoglycerides and fatty acids are recombined here in order to form triglycerides, and then they're combined with lipoproteins and cholesterol in the Golgi body in order to produce what's known as chylomicrons. These chylomicrons are vesicles which move to the cell membrane, fuse with the cell membrane, releasing their contents by exocytosis, meaning that the triglycerides can then enter the lacteal. Last up, what's the role of the large intestine? Its role is to absorb water. To study lipid digestion, a scientist placed a tube into the gut of a healthy 20-year-old man. The end of the tube passed through the stomach but did not reach as far as the ileum. The scientist fed the man a meal containing triglycerides through the tube. The scientist also used the tube to remove samples from the man's gut at intervals after the meal. The scientist measured the type of lipid found in the samples. Some of her results are shown in the table below. Sample A, time of collection after the meal is 45 minutes. The concentration of fatty acids is 2.7. The concentration of triglycerides is 0.6. In sample B, it's a much longer time after the meal. We can see that the concentration of fatty acids has increased and the concentration of triglycerides has decreased. Do remember from an earlier module that a triglyceride consists of 3 fatty acids and 1 glycerol. So over time, we're expecting that triglyceride has been broken down into fatty acids and glycerol, hence why you see the concentration has decreased here whilst the concentration of fatty acids has increased. Use your knowledge of lipid digestion to explain the differences in the results for samples A and B shown in the table above. You should assume that no absorption has occurred. So what we're going to say is that the concentration of triglycerides decreases, and the reason for that is the action of the enzyme lipase, which digests the triglycerides to fatty acids and glycerol. Which causes triglycerides to be hydrolyzed to fatty acids and glycerol. And then to get that third mark, just point out that hydrolysis really means that it's the ester bonds between the fatty acids and the glycerol which are breaking. After collecting the samples, the scientist immediately heated them to 70 degrees for 10 minutes. Explain why. As soon as we talk about high temperatures, we need to talk about enzymes. So high temperatures denature the lipase enzyme. So no further digestion or hydrolysis can take place. Describe the role of micelles in the absorption of fats into the cells lining the ileum, 3 marks. So remember that micelles are small droplets formed of monoglycerides, fatty acids and bile salts. They're responsible for bringing those monoglycerides and fatty acids to the lining of the ileum, and they make those fatty acids more soluble in water. Describe the role of enzymes in the digestion of proteins in a mammal. So let's talk about a variety of enzymes, including endopeptidases, exopeptidases, dipeptidase, which are all responsible for the hydrolysis of peptide bonds at various points along the polypeptide. A variety of enzymes hydrolyze peptide bonds in polypeptides, including endopeptidases, which act in the middle of the polypeptide, exopeptidase, which act at the end of the polypeptide, producing amino acids, and dipeptidase act between 2 amino acids, producing single amino acids. Scientists investigated how the diet of rabbits affected their digestion and the absorption of protein. The scientists fed rabbits an identical mass of food but varied the percentage of protein in the food. The scientists measured the mean mass of protein fed to the rabbits that was absorbed, which they then expressed as a percentage value. The scientists' results are shown in Figure 1. The error bars show plus or minus 2 standard deviations. Plus or minus 2 standard deviations cover 95% of the data. So remember, standard deviation is a measure of how spread out the data is. The greater the standard deviation, the greater the spread. If we look at the results here are error bars. We're always looking to see if they're overlapping or non-overlapping. Now, if they overlap, such as the case here, you can see those error bars clearly overlapping. It means that there is no significant difference, which makes it very hard to draw conclusions from the data. So what can you conclude about the absorption of the products of protein digestion as the percentage of protein increased in the rabbit's food? So here it is increasing. Here's the mean percentage of ingested protein absorbed. So you can see that we really need to understand what standard deviation means, because I'm going to start by saying that there's no significant difference in the protein absorption as you increase the percentage of protein in the diet. And the reason for that is because the standard deviation error bars overlap. And what that really means is that the mean percentage of ingested protein absorbed is not affected by the percentage of protein in the diet. The digestive system of a rabbit is shown in Figure 2. The food eaten by a rabbit is digested mainly by microorganisms in its cecum. The cecum is a section of intestine attached between the ileum and the large intestine. The resulting semi-digested material leaves the anus of a rabbit as soft cecal droppings. The rabbit then eats these cecal droppings. Use this information in Figure 2 to suggest how eating its own cecal droppings helps a rabbit's digestion and absorption of dietary protein. It does sound like quite an unpleasant thing that it's doing, but after all, it only really eats vegetation, grass. So you can see by eating those cecal droppings, more of that undigested protein will be broken down, because that protein is going to pass again through the stomach and the ileum, meaning that more amino acids overall can be absorbed. Eating cecal droppings means that more of the remaining protein can be broken down as protein passes through the stomach and ileum. Again, this means that more amino acids are absorbed. Discussing all things now relating to the heart and circulatory system. So why do multicellular organisms require transport systems? Well, we previously talked about the fact that unicellular organisms have a much larger surface area to volume ratio, meaning that diffusion occurs nice and quickly. With larger multicellular organisms, their surface area to volume ratio is much smaller, so diffusion over long distances doesn't occur fast enough. So as well as overcoming those difficulties, the transport systems will obviously be used to transport substances. These include hormones and enzymes, which need to travel to a different region away from where they're made. Food absorbed at the small intestine needs to be transported to cells where it is used in metabolic reactions. And waste products such as urea and carbon dioxide need to be taken to excretory organs such as the lung excrete carbon dioxide, the skin excretes sweat, and our kidneys excrete urine. So the reasons why multicellular organisms require transport systems is twofold. Yes, they need to transport various substances, but it's also largely due to their small surface area to volume ratios. We're going to be looking at one substance to begin with, which is oxygen. Oxygen is transported by our red blood cells, which we call erythrocytes. The oxygen binds to a pigment found within those red blood cells, which is known as haemoglobin. Haemoglobin is an example of a globular protein made up of 4 polypeptide chains. This is its quaternary structure, and each of these polypeptide chains contains the heme prosthetic group. Remember that prosthetic group is a group in addition to the normal elements that you find within an amino acid. That heme prosthetic group contains iron, and that's why it's important that we have diets that contain plenty of iron in order to give us strong blood. It's in order to make the haemoglobin in our red blood cells. Now the oxygen binds to haemoglobin, forming oxyhaemoglobin. We'll now talk in great detail how oxygen is loaded onto that haemoglobin and how it's removed, and this is largely due to the partial pressure of oxygen in a particular environment. So that could be respiring cells, it could be in our atmosphere. You'll meet partial pressures a lot in Year 2 chemistry, but what partial pressure refers to is the contributing pressure of a single gas to the total pressure of a mixture of gases. So in simple terms, here in biology, we're really thinking, what is the partial pressure of oxygen? How much oxygen is present in a particular place? So if we start in the lungs, there'll be a high partial pressure of oxygen present in the alveoli, whilst there is a much lower partial pressure of oxygen in the capillaries, and as a result, oxygen moves from the alveoli to the capillaries by diffusion. At respiring tissues, the opposite is true, so our respiring tissues will contain a low partial pressure of oxygen. The blood will contain a comparatively higher partial pressure of oxygen, and therefore oxygen diffuses into the respiring tissues, which makes sense. After all, they need to carry out respiration, which requires oxygen. Now, the pigment haemoglobin has what's known as a high affinity for oxygen. It wants to bind to that oxygen, and we know it's going to bind very readily to oxygen in the alveoli due to the high partial pressure of oxygen found there. As we know, oxyhaemoglobin is formed, and then we know that oxygen will be released from the haemoglobin in respiring tissues which contain a low partial pressure of oxygen. Now, the way in which oxygen binds to haemoglobin is incredibly clever. When the first molecule of oxygen binds to haemoglobin, it causes a conformational change, a change in the tertiary and quaternary structure of the haemoglobin. By doing this, it exposes a second binding site for oxygen. This increases the haemoglobin's affinity for oxygen, making it easier to load on that second oxygen molecule. Then the opposite is true when we reach respiring tissues. When we offload that first oxygen molecule, another conformational change occurs. The tertiary and quaternary structure of the haemoglobin alters, and its affinity for oxygen decreases. And by doing so, it makes it easier for the next oxygen molecule to leave. So if we look at the oxygen dissociation curve for human haemoglobin, you can see bottom left, at low partial pressures of oxygen, there are few heme groups bound to the oxygen, and haemoglobin therefore does not carry much oxygen. As we reach the steeper portion of the graph, where the partial pressure of oxygen increases, we can see that more heme groups are bound to oxygen due to that conformational change, making it easier for further oxygen molecules to bind. So there's a higher percentage saturation of haemoglobin with oxygen, and then we reach higher partial pressures still, and the haemoglobin becomes saturated with oxygen. The significance of this saturation curve means that at high partial pressures, such as in the alveolus, haemoglobin gets rapidly loaded with oxygen, and then when we see a small drop in partial pressure of oxygen, due to the shape of that curve, you can see a rapid unloading of the oxygen and a desaturation of the haemoglobin. This means that oxygen is free to diffuse into the cells where it's needed in respiration. You'll have seen different types of this oxygen dissociation curve. What happens if you move that curve, that sigmoidal curve, to the left? Think about what that means. It means that the haemoglobin has a greater affinity for oxygen, meaning that it loads oxygen more readily but unloads less readily. If the oxygen dissociation curve moves to the right, then that haemoglobin has a lower affinity for oxygen, meaning that it unloads more readily but loads with oxygen less readily. So in a growing fetus in the mother's womb, you'll find that the fetal oxygen dissociation curve is on the left of the adult. This means that fetal haemoglobin has a higher affinity for oxygen, which means that its haemoglobin loads more readily with oxygen but unloads less readily. So why does the fetal haemoglobin curve sit towards the left of the adult haemoglobin curve? It's because the placenta, which is an organ supplying the growing fetus, has a comparatively low partial pressure of oxygen. Adult haemoglobin will therefore release oxygen at the placenta. The fetal haemoglobin has a higher affinity at lower partial pressures found at the placenta, meaning that fetal haemoglobin is still able to take up oxygen at the comparatively low partial pressures of oxygen found at the placenta. Carbon dioxide is a gas which also has an effect on haemoglobin, and at higher partial pressures of carbon dioxide, so in effect higher concentrations of CO2, haemoglobin will give up oxygen more easily. And again, this makes sense because respiring cells will produce carbon dioxide, and therefore that higher partial pressure of carbon dioxide will prompt more oxygen unloading. The effect of carbon dioxide is to shift the oxygen dissociation curve to the right, and we call this the Bohr shift or the Bohr effect. So to reiterate, the Bohr effect is very important because it has the following effects. We've already said that actively respiring cells will produce lots of CO2, meaning that there's a higher partial pressure of carbon dioxide at respiring tissues. This means that haemoglobin gives up oxygen more readily. Comparatively speaking, the lungs have a lower partial pressure of carbon dioxide, and if you look at that graph, you'll see that this will have the effect of causing haemoglobin to bind to oxygen more readily. So if we have a look at these notes from my revision guide, we can see the benefits of the Bohr shift to actively respiring tissue. First up, actively respiring tissue, as we know, needs more oxygen for aerobic respiration. We know that it will also be producing more carbon dioxide, and since haemoglobin is involved in the transport of carbon dioxide, it means that there will be less haemoglobin available to combine with oxygen. So the Bohr shift therefore causes more oxygen to be released. What effect does pH have on haemoglobin's affinity for oxygen? Well, as the pH decreases, so it becomes more acidic, there are more hydrogen ions present, haemoglobin's affinity for oxygen decreases. So we know that the Bohr effect occurs, but why does it occur? Let's combine everything I've just mentioned. So we know that an increased partial pressure of carbon dioxide reduces the affinity of haemoglobin for oxygen. But why is that? Well, increased carbon dioxide increases the acidity. The hydrogen ions interact with haemoglobin to form haemoglobinic acid. In this way, the haemoglobin's acting as a buffer. It's helping to reduce changes in pH brought about by the increased carbon dioxide, and the hydrogen ions present displace the oxygen in haemoglobin, and this means that more oxygen is released where respiration is occurring, because after all, that releases CO2. Make sure you can apply everything you've learned to organisms that live in different environments. Think about what their oxygen requirements will be, how the partial pressures will be affected. So if we were to compare the adult human dissociation curve with an organism that lives in mud or soil or somewhere at high altitude where there's lower partial pressures of oxygen, these organisms will have oxygen dissociation curves which are shifted to the left. Remember, this means that their haemoglobin will have a higher affinity for oxygen, so oxygen will be loaded on more readily but will be unloaded less readily. Binding of one molecule of oxygen to haemoglobin makes it easier for a second oxygen molecule to bind. Explain why. This is quite a nice, straightforward question. So remember, the binding of the first oxygen molecule changes the tertiary structure of haemoglobin, leading to the uncovering of another oxygen binding site, and that's actually enough for the 2 marks. The graph shows the oxyhemoglobin dissociation curves for fetal hemoglobin HbF and adult hemoglobin HbA. So on the y-axis we have the percentage saturation of hemoglobin with oxygen, the x-axis the partial pressure of oxygen. Explain how changes in the shape of the hemoglobin result in the S-shaped sigmoid oxyhemoglobin dissociation curve for the adult hemoglobin. So what changes take place in the shape of the hemoglobin? Well, remember, when the first oxygen binds to the hemoglobin, it causes a change in that tertiary structure, meaning that it uncovers more binding sites, allowing the second oxygen molecule to bind more easily. At birth, 98% of the hemoglobin is fetal hemoglobin. By the age of 6 months, the fetal hemoglobin has usually completely disappeared from the baby's blood and has been replaced by adult hemoglobin. Use the graph above to explain why this change is an advantage for the baby. So we're replacing the fetal hemoglobin with the adult hemoglobin. So if we look down here at a low partial pressure of oxygen, you can see that the adult hemoglobin, which would be representative of tissues which contain lots of CO2 and low oxygen concentrations. You can see that the adult haemoglobin has a lower percentage saturation with oxygen, which means that oxygen will be more easily unloaded, which is sensible because that's needed in aerobic respiration. So the simplest way of saying that is that the adult haemoglobin, HbA, has a lower affinity for oxygen at low partial pressures, meaning that oxygen can be more easily unloaded and used in respiration. Sickle cell disease is caused by production of faulty adult haemoglobin. This results in a reduced ability to transport oxygen to tissues. Scientists investigated the use of a substance called hydroxyurea to treat babies with sickle cell disease. Hydroxyurea changes the concentration of fetal haemoglobin in the blood. The scientists carried out an investigation with 122 babies who had sickle cell disease. Each baby was given hydroxyurea for 41 months. The scientists then found the mean change in the concentration of fetal haemoglobin in the baby's blood. So we can see that after treatment with the hydroxyurea, the mean concentration of fetal haemoglobin has increased. The scientists concluded that treatment with hydroxyurea would increase the concentration of oxygen in the blood of babies with sickle cell disease, suggest how the graph and table support this conclusion. So we can already point out what I've already said, which is that there is a significant increase in fetal haemoglobin after treatment. And then if we use the graph, remember that the fetal haemoglobin, as you can see here, has a higher affinity for oxygen, and that therefore means that more oxygen can be carried. Let's talk about the structure of the heart. I'm going to give you some good tips as to how you can get to grips with this super quickly. We'll start by drawing the heart as a box so that we can label the associated blood vessels and chambers, and then I'll apply everything we've just learnt to a more realistic diagram of the heart, and you'll see how straightforward this can be. So remember, the heart has 4 chambers. The sides of the chambers are flipped, so that means that this side over here is the right side of the heart, this side is the left side. So to label them, the entrance chambers of the heart are known as atrium. So there's your right atrium, your left atrium, your right ventricle, and there's your left ventricle. Remember that arteries carry blood away from the heart. Try and remember the 2 A's go together, whereas veins bring blood back to the heart. A few extra points to make is that the left side of the heart contains oxygen, it's oxygenated. The right side is deoxygenated. And then I want you to remember that pulmonary, the word pulmonary just means relating to the lungs. So now we can start labeling various blood vessels. The blood flow is in this direction, so it comes into the atria. They contract, forcing blood into the ventricles, and then the ventricles contract, forcing blood out of the heart. So what are the name of these various vessels? Well, let's think about it. If the left side is oxygenated, we know that blood must have come from the lungs. After all, that's where oxygenation occurs. We know that the vessel coming into the heart must be a vein. So the name of this vessel is the pulmonary vein. Then down here, you know it must be an artery because it's a vessel leaving the heart. It's the main artery, it's oxygenated, it's the aorta. The blood goes around the body and becomes deoxygenated due to the cells using the oxygen in respiration. The blood has to return to the heart before going off to the lungs for oxygenation. What is the name of this vessel? It's coming from the body. It must be a vein because it's coming towards the heart. It is the vena cava. And then down here, this blood is leaving the heart, so it must be an artery. Where's it going? Well, it needs oxygenating, so it must be the pulmonary artery. So practice drawing your box diagram to make sure that you're happy with the basic structure of the heart before we turn to the more anatomically correct version. So here's a more anatomically correct version of the heart, but in essence it's the same. We have our 4 chambers. Let's start by labelling them. This remains the left side of the heart. This is the right hand side of the heart. So the left atrium, left ventricle, right atrium, right ventricle. Just keep an eye on which way that blood is flowing. So if the blood comes in here, it's going to come in via the vena cava. It's deoxygenated blood entering via a vein. The blood flows into the right atrium, into the right ventricle, and then it needs to be oxygenated at the lungs. This vessel is leaving the heart, so it must be an artery. So this is the pulmonary artery. The blood becomes oxygenated. It needs to enter the heart so it can be pumped onto the rest of the body. It enters the heart here via the left atrium. Because it's coming from the lungs, again it's pulmonary, but because it's entering the heart, it's the pulmonary vein. So the blood flows into the left atrium, down to the left ventricle, and then out of this huge vessel here that has a very characteristic arch with huge vessels coming off it. It's the main artery of the body. It's therefore the aorta. A couple of things to notice. Look, the wall of the left ventricle is much thicker than the right. It has far more muscular tissue. The reason for that is because it has to pump blood at a very high pressure, because that blood's travelling much further. After all, it's going all around the body, compared with here, where the blood is just being pumped to the lungs. We've got a variety of valves present, which help to ensure that blood flows in the correct direction. Over here, you have between the left atrium and the left ventricle, the left atrioventricular valves, otherwise known as the bicuspid valves. And then over here, you have the tricuspid or right atrioventricular valves, and they prevent backflow of blood into the atria. And then at the base of the arteries, so both the pulmonary arteries and the aorta, you have the semilunar valves, which prevent backflow of blood into the ventricles. So do practice this yourselves. Either go back in my video to when this was unlabelled, have a go at labelling it yourself, and then see if it matches my answer, or find another one online, but do practice labelling the heart. And then the human circulatory system, that really means the name of the various blood vessels flowing into and out of the organs of the body. I promise this diagram isn't as awful as it looks. Always start with the heart, so get your chambers labelled and make sure that you're happy with the various blood vessels coming into and out of each chamber. There's the pulmonary vein feeding oxygenated blood from the lungs into the left atrium. The aorta is the main artery of the body, taking oxygenated blood down a series of arteries. Do be aware that if you have anything relating to the liver, we use the word hepatic. So because that vessel here is coming off the aorta, it is an artery, it's feeding into the liver, it is the hepatic artery. The blood vessel carrying deoxygenated blood away from the liver is the hepatic vein. That's going to feed into the vena cava, which goes into the right atrium. You've got a special vessel known as the hepatic portal vein. This is a vessel containing high concentrations of glucose due to the fact that it links the stomach and intestines to the liver. The word renal means relating to the kidneys. That's why the blood vessel supplying oxygenated blood to the kidneys is the renal artery. The blood vessel taking deoxygenated blood away from the kidneys is the renal vein. So the vena cava takes that deoxygenated blood to the heart so that when that right ventricle contracts, that blood can flow into an artery, and it is the pulmonary artery because it's going to the lungs. Remember, most arteries in the body are oxygenated. The exception is the pulmonary artery. Most veins are deoxygenated. The exception is the pulmonary vein, transporting oxygenated blood from the lungs to the heart. So if we look in super close detail then in terms of how blood flows through the heart, we know that oxygenated blood is coming from the lungs. The blood flows through the pulmonary vein into the left atrium. The left atrium contracts, forcing blood through the bicuspid valves into the left ventricle. The left ventricle contracts, forcing oxygenated blood through the semilunar valves into the aorta, meaning that oxygenated blood can now flow around the body. The blood returns to the heart, this time deoxygenated into the right side via the vena cava. The right atrium contracts, forcing blood through the tricuspid valves. The blood enters the right ventricle, and then you have the contraction of the right ventricle, meaning that blood flows through the semilunar valves into the pulmonary artery, where it then goes off to the lungs for reoxygenation. The septum, which is a wall of muscle right here, separates the left and right hand sides of the heart, meaning that oxygenated blood and deoxygenated blood don't mix. Now that's important because after all, you don't want deoxygenated blood being delivered to the body, because after all, the respiring cells need that oxygen. Secondly, you don't want oxygenated blood returning to the lungs. After all, the lungs is the organ of oxygenation. But why are the walls of the ventricles thicker than that of the atria? And that's because the atria up here receive blood with lower pressure. And secondly, they only need to pump the blood a very short distance into the left and right ventricles, whereas the ventricles have to contract strongly in order to pump high pressure blood to either the lungs or rest of the body. Let's have a little look at my revision guide available on the website. So we're looking at the difference here between an open and closed circulatory system. Now, an open circulatory system found in insects has very few transport vessels. It has something known as hemolymph instead of blood. The hemolymph doesn't carry oxygen or carbon dioxide, which is obviously something which is characteristic of our blood. Hemolymph is pumped straight from the heart into the body cavity at low pressure, and it comes in direct contact with cells. And as I said, that open circulatory system is found in insects. Whereas a closed circulatory system, closed indicates the fact that the blood is enclosed in blood vessels, such as veins and arteries. The blood doesn't come in direct contact with cells of the body due to those blood vessels. The heart pumps the blood through vessels under high pressure. And how do our cells receive the oxygen? By diffusion. And as I said, closed circulatory systems are found in mammals. You'll have met the single and double circulatory system at GCSE and IGCSE. The single circulatory system refers to the fact that the blood passes through the heart once for each circuit of the body, and it passes through two sets of capillaries. Oxygen and carbon dioxide are exchanged with the environment in the first set of capillaries, whilst nutrients and gases are exchanged with cells in the second, and we find a single circulatory system in fish. Whereas the double circulatory system is characterized by the blood flowing twice through the heart for every once around the body. Remember, we said it goes into the left atria via the pulmonary vein, into the right atria via the vena cava. So you have two types of circulatory system in that double circulatory system. The pulmonary circulation, remember pulmonary refers to lungs, refers to blood going from the heart to the lungs back to the heart. Whereas the systemic circulation takes that blood from the heart all around the body and then returns it to the heart, such as you find in mammals. And then another set of vessels which require special mention are the coronary arteries. These are vessels which snake over the outside surface of the heart, providing the heart with its own oxygen requirements. After all, the heart is an incredibly hard-working organ which must contract continuously throughout our lifetime. The coronary arteries help maintain a regular heart rhythm by supplying glucose and oxygen to the heart, which is then used in respiration, and then the ATP from respiration is used to contract the heart muscle with a regular rhythm. Now, cardiac muscle refers to muscle belonging to the heart, so it has various characteristics. First of all, it's myogenic, and that means that it can contract automatically. As I said, it's essential that our heart continues to contract throughout our lifetimes. Cardiac muscle also contains cardiac arteries. These supply oxygen and nutrients to the heart, as well as valves which prevent the backflow of blood. Now we want to look at the cardiac cycle, which is a sequence of contractions and relaxations of the heart muscle that occurs during one heartbeat. Now this is in effect a graph showing the pressure in millimeters of mercury. And these 3 lines represent various pressures within the heart. What's key to note is that the maximum pressure in the atrium is always lower than the maximum pressure in the ventricles, and that's obviously due to there being less muscle in the walls of the atria. So where you see this increase in pressure, this is what we know to be the atrial systole, which is when the atrium contracts. That will force the blood into the ventricles, and therefore the atrioventricular valves, or the bicuspid and tricuspid valves, close because the blood has left the atrium and we want to prevent backflow into the atria. You then have your ventricular systole, because after all the ventricles are filling with blood. The filling of the ventricles will force blood into the aorta, hence why your semilunar valves in the base of your aorta open, meaning that pressure builds up in the aorta as blood flows into it. And then to prevent further backflow, the semilunar valves close, meaning that no more blood can flow into the aorta, hence why you see a decrease in pressure here. And then when your ventricular pressure drops below atrial pressure, the atrioventricular valves open, meaning that blood entering the atria can then flow into the ventricles. And then lastly, before we leave the cardiac cycle behind, we're explaining how pressure changes in the heart bring about the closure of those atrioventricular valves. You do need to know the anatomy of the heart extremely well so that you actually know what we're talking about. Remember, these are the valves between the atria and the ventricles. So your ventricular systole, so when the ventricles are contracting, raises our ventricular pressure higher than that of the atrium. And I've just drawn this diagram here. I've reverted back to my box diagram and I'm just showing you the left side of the heart. So what we're saying here is that when the pressure within the left atrium is greater than the pressure in the left ventricle, obviously blood will be forced into the left ventricle. And because of the shape of those valves, it does mean that those valves remain open. However, as the left ventricle pressure increases greater than that of the left atria, it's going to force these valves to shut, meaning that no backflow of blood into the left atrium can occur. And the poorly drawn dotted lines refer to valve tendons, which prevent the inversion of the valves. And all this bit here means is ventricular systole refers to ventricular contraction, which will obviously lead to an increase in ventricular pressure, which explains why the pressure in our ventricles is higher than the atrial pressure. And then lastly, why does the aortic pressure, so remember that red line up here, remain high throughout the cardiac cycle? And that's due to the presence of thick muscle and elastic fibres in the artery wall. Some key definitions relating to the heart, so starting with the cardiac output. Simply put, this is the volume of blood the heart pumps per minute. Now we need to discuss the stroke volume. This is the volume of blood pumped out of each ventricle per heartbeat. The heart rate is the number of beats per minute. And there's a simple calculation to find out cardiac output. It's stroke volume multiplied by heart rate. So let's take a look at this graph, which is showing blood pressure in the circulatory system. We're comparing arteries, including the aorta, with capillaries and veins. We have blood pressure here on the y-axis in kPa. So the first thing to note is the aorta has the highest pressure, and that makes sense, doesn't it? Because the aorta is closest to the heart. So obviously, whether the heart is contracting or relaxing, so whether there's systole or diastole, will have the largest impact on the aorta. After all, the aorta feeds out of the left ventricle. So when the ventricles are contracting, forcing blood into the aorta, you'll expect high blood pressure, and then when the ventricles relax, you'll see a decrease in blood pressure found within the aorta, which is seen here in this trough when diastole is occurring. Do notice as well that as we move along the x-axis, these vessels are getting further away from the heart, and that therefore explains why you'd expect a decrease in blood pressure. The other thing to notice is these large fluctuations in blood pressure, especially when we compare them to the capillaries and veins, and it's these huge changes in blood pressure which actually go some way to explain the structure of our arteries, including the aorta. Remember, their walls contain large quantities of elastic fibre, and that enables the vessels to expand and recoil based on whether the heart is contracting or relaxing. So to reiterate, we can see the pressure drops as the distance from the heart increases. Remember, it's the veins which will return blood to the heart. There's the greatest pressure drop whilst the blood is in the arteries, compared with at the veins and capillaries where the pressure is largely constant. And then to explain what causes the overall change in pressure as the blood flows from the aorta to the arteries to the capillaries, this is to do with the total cross-sectional area. Obviously, capillaries, we know that they are tiny, but the huge quantity of them means that the blood has far more routes, in effect, to flow through. And so what we would really say is that the total cross-sectional area of the capillaries is greater than the arteries, and that explains why there's a drop in pressure. And I'm just going to pause here so you can have a look at my revision guide. So I've really spelled out exactly what you can take away from that graph in terms of the sorts of notes you'll need to be able to reproduce in the exam. You might have heard of ECGs before. ECG stands for electrocardiogram. Now, the purpose of an ECG is to help us diagnose heart problems, and it does that by measuring the electrical differences in the skin, which we say is the equivalent to the electrical activity of the heart. So here's a normal ECG trace, and here you can see exactly 2 heartbeats being shown. If we look at the structure of our blood vessels, so that is our arteries, veins and capillaries, we're going to link their structure together with their function. So starting with the arteries, the first thing to notice is this comparatively narrow lumen. Remember, the lumen is the space through which the blood flows. Surrounding that narrow lumen, you have thick walls that contain 3 layers. There's thick, smooth muscle and elastic fibres, and that needs to withstand the high pressure of the blood flowing through that lumen. The narrow lumen further maintains the high pressure within the arteries. The function of the arteries, remember, is to carry blood at high pressure from the ventricles of the heart to other tissues of the body. We look at the veins now. Comparatively speaking, they have a much wider lumen. Their walls again consist of 3 layers, but this time they have much thinner smooth muscle and elastic fibre walls. The reason for this is twofold. First of all, the pressure of the blood flowing through that lumen is so much lower, so they don't need to be thick in order to withstand high pressure. Secondly, the thin walls mean that the veins can be pressed flat by muscles to carry blood under low pressure. If we look at a side view of a vein, so here's our blood vessel, so here's our vein, they have valves. And so if the blood is flowing in this direction, you can see how those valves will help prevent the backflow of blood. Now, the purpose of the veins is to carry blood at low pressure from the tissues of the body back to the atria of the heart. And then last up, the capillaries. Now, the first layer of these blood vessels, the kind of layer that surrounds the lumen, is known as endothelium. So our capillaries have a single layer of endothelium. They have an extremely narrow lumen. Remember, capillaries are very, very small. They have thin walls, which are only 1 cell thick. Those walls are permeable to enable exchange of materials between cells in the tissue and the blood in the capillary. The purpose of the capillaries is to carry blood through the tissues. And again, here's a nice summary from my revision guide. So we mentioned the presence of the elastic fibres. Specifically, their role is to stretch and recoil in order to make the blood vessel walls flexible, whereas the smooth muscle, which is present as it's a muscle, it can contract and relax in order to change the relative diameter of the lumen. If you look at this label here, we can see that there's a layer of collagen, so that third layer present in the walls of these vessels. And collagen, remember, is a protein which provides structural support to the vessels. One last vessel to mention is the arterioles. These link the arteries and capillaries. In terms of their structure, their walls contain smooth muscle, elastic fibres. They have more smooth muscle compared with arteries and fewer elastic fibres compared with arteries. And they also have a smaller lumen compared with arteries. If we really focus in on the capillaries, remember that their function is the exchange of substances. In terms of their structure, I already mentioned, but I might as well pop it here in the spider diagram. They have a narrow lumen, thin walls. They have an endothelial layer lining the lumen. They have permeable walls. And in terms of their adaptations for this function, they have a large surface area for diffusion. Touching on that point I made previously, their total cross-sectional area is larger than that of the arteries and even the arterioles. And what that does is it slows down the blood to give more time for exchange. The narrow lumen means that the red blood cells pass through one at a time, slowing down the blood. So red blood cells pass along one cell at a time. So more time for the exchange of substances. And the fact that they have such thin walls provides a short diffusion pathway. Let's mention venules now. These link capillaries to veins. They have very thin walls with very little smooth muscle. They allow blood to flow into the veins. Notably though, they have no valves, so they can't control the direction the blood flows. We're now going to compare blood, tissue fluid and lymph, paying particular attention to the components of these three fluids. So we'll be looking at the various cells carried by them, the dissolved solutes, proteins, as well as the location of these fluids. Starting with the one you'll be most familiar with, blood. So what are the components of blood? You've got red blood cells, erythrocytes, which obviously transport oxygen. You've got white blood cells. You've got plasma, which transports dissolved solutes such as amino acids, sugars, fatty acids, carbon dioxide. Other components include proteins, which include clotting factors, hormones, plasma proteins, and immunoglobulins. And other components include those all-important platelets, which help to clot the blood at the site of a wound. So an awful lot of components carried by our blood. Be prepared to talk about which direction all these substances are moving in. So obviously oxygen is being transported to respiring cells. Carbon dioxide is being transported away from respiring cells. You've got digested food molecules like glucose and amino acids, which will be absorbed through the lining of the small intestine into the blood there. Don't forget you've also got urea, which is produced by the breakdown of excess protein. Remember, deamination occurs in the liver. The hormones will be transported from the glands where they're made to the target organs where they act. And the key thing to remember is that blood is carried inside blood vessels. Let's move on to tissue fluid now. What is tissue fluid? It's fluid forced out of blood into surrounding space around cells. So tissue fluid isn't found within blood vessels, it's found bathing the cells of the tissues. In terms of the components of tissue fluid, you've only got a few white blood cells, and there'll be no red blood cells or platelets. In terms of dissolved solutes, those will include amino acids, sugars and fatty acids, because the tissue fluid has a huge role to play in delivering amino acids, glucose, fatty acids to cells. If we think about the quantity of oxygen and carbon dioxide found in tissue fluid, there'll be less oxygen and more CO2, and the reason for that is because the tissue fluid delivers oxygen to respiring cells and removes carbon dioxide from respiring cells. So we know that tissue fluid is fluid forced out of blood into the surrounding space around the cells, but how is that fluid forced out of the blood? Well, what feature of the capillaries allows that fluid to be forced out of the blood? It's the fact that the capillaries have small gaps between their epithelial cells. And because those gaps are so small, it makes sense that larger molecules can't leave the capillaries, so that includes the red blood cells, most of the white blood cells, and plasma proteins. So that accounts for the fact that there are very few white blood cells, no red blood cells present in tissue fluid. So red blood cells and most white blood cells and plasma proteins are too large to leave the capillary because they can't squeeze through the gaps between those epithelial cells, and therefore they remain in the blood. So just have a look at the larger picture. Tissue fluid is the fluid forced out of blood into the surrounding space around cells. Remember, the role of the tissue fluid is to deliver oxygen to respiring cells, remove CO2 from respiring cells, and to deliver amino acids, glucose, and hormones to cells. These molecules and substances are small enough to squeeze through the gaps between those epithelial cells. They're able to leave the capillaries in order to form the tissue fluid which bathes the cells, whereas larger proteins are unable to squeeze through those gaps. And we'll now look in greater detail how that tissue fluid is formed. So we now need to consider in detail how that tissue fluid is formed. So make sure you're paying lots of attention here. Remember that the blood feeding into the capillaries comes from the arterioles, and as such has relatively high pressure due to the fact that the blood supplying the arterioles has come from the arteries, which are obviously filled by the contraction of the ventricles in the heart. So I'm really going to spell this out. The ventricles contract, forcing blood into the arteries, then onto the arterioles, then into the capillaries, and as such that blood is at quite high pressure really. And what do we call that pressure? We call it hydrostatic pressure. So I'll write a definition of hydrostatic pressure now. This is pressure from the heartbeat, and as a result of that pressure, you find that liquid from the blood is forced out through gaps between cells of the capillary lining. So you can immediately see why we say that tissue fluid bathes the cells. This is in effect liquid which has been forced out of the blood in the capillaries. And what will that liquid contain? Gases such as oxygen, various nutrients like glucose, as well as water from the plasma. And then if you think about it, because we force so much fluid out of the capillaries, as a result the pressure decreases. So pressure decreases inside the capillary due to the loss of this liquid. This has a knock-on effect of meaning that lots of tissue fluid can return to the capillary, this time at the venous end of the capillary, by osmosis. And do remind yourself of the definition of osmosis. Remember, it's the movement of water from an area of high water potential to an area of low water potential. So at the venous end, it's this osmotic pressure that is more important compared with the hydrostatic pressure. And why this is so useful is because when that fluid moves back to the capillary, this time it's going to take things like carbon dioxide, which have been produced by respiring cells. And by entering the capillaries and then moving on to the venules and then the veins, it means that carbon dioxide can be taken back to the heart for final removal at the lungs. So I'm just showing you the route that blood is taking. So if you think about it, the formation of the tissue fluid is super helpful for the body because it's a great way of ensuring that oxygen, various nutrients, water, etc. are delivered to the cells where it's needed. And then due to that decrease in pressure, the fact that that tissue fluid can then return to the capillary, and it's around 90% of that tissue fluid which returns, taking with it that carbon dioxide which has been made by respiring cells that can then be returned through the system of the venules, veins, heart, and finally to the lungs for excretion. But remember, that fluid moves back due to osmosis, and this is oncotic pressure as opposed to hydrostatic pressure. And what happens to the remaining 10% of fluid is that it's used to produce lymph, which we'll talk about shortly. So just to show you this diagram, so this is the arterial end of the capillary, here's the venous end. So we know that the pressure at the arterial end is fairly high because it's come from the arterioles, the arteries, and therefore that pressure is dictated by the heartbeat, so we call that hydrostatic pressure. This means that fluid containing gases like oxygen needed for respiration, glucose, water leaves the capillary at the arterial end, forming tissue fluid which bathes the cells of the body. And then as that fluid's removed, clearly the hydrostatic pressure will decrease, and as a result, fluid is returned to the capillary at the venous end, this time by osmosis. We call this the oncotic pressure, which is the tendency of water to move from a region of high water potential to one of low water potential. And so 90% of the fluid returns to the capillary, leaving 10% to form lymph. Third substance now, which is lymph. First of all, what is lymph? It's 10% of tissue fluid that does not return to the capillaries. The lymph is really what's known as the lymph system, which consists of both lymph vessels as well as lymph nodes. The lymph vessels are a series of blind-ended tubules where valves prevent the backflow of the lymph. Whereas the lymph nodes are in effect small nodules which contain large quantities of lymphocytes. So that white blood cell, which is very important in our immune response, they produce antibodies which kill pathogens. And so really that is the role of our lymph, to contain lots of lymphocytes which produce antibodies upon infection with a pathogen. And those antibodies will have to enter the blood. So in terms of the components of lymph, as we've already said, there'll be many white blood cells, specifically lymphocytes. You'll have no red blood cells or platelets. In terms of dissolved solutes, there'll be few amino acids and sugars, but there'll be lots of fatty acids. Let's understand why now. What is the source of the fatty acids in the lymph? Well, remember these are absorbed at the small intestine. If you remember the structure of the villi, there are those special structures known as lacteals which absorb the fatty acids which can then be transferred to the lymph system. One last thing to be aware of is that lymph can return to the blood, and that's in structures known as subclavian veins. So to reiterate, the lymph is a special portion of tissue fluid. It's 10% of tissue fluid that does not return to the capillaries. The lymph system consists of both lymph vessels, which are blind-ended tubules containing valves that prevent backflow of lymph. The lymph nodes contain a very high number of lymphocytes which will produce the antibodies needed to kill pathogens. In terms of the components, yes, you'd expect lots of white blood cells. You won't get red blood cells or platelets in the lymph. You will get large numbers of fatty acids, and that's because those fatty acids, when they're absorbed at the small intestine, enter the villi, specifically the lacteals, where they can then move across to the lymph system. The graph shows the volume changes in the left ventricle of a human heart during 2 cardiac cycles. The numbers 1, 2, 3 and 4 represent times when the heart valves open or close. On the y-axis we have volume of ventricle, x-axis time in seconds. Use information from the graph to complete the table in part A. Place the number 1, 2, 3 or 4 in the appropriate box. Obviously we need a good working knowledge of both the cardiac cycle as well as the structure of the heart. So just to remind you over here with my box diagram, we've got the right atrium, right ventricle, left ventricle, various blood vessels leading into the heart and away from the heart. So this is your vena cava, your pulmonary vein, your pulmonary artery and your aorta. And then I just want to label various valves, making sure we're looking at the semilunar valves and the atrioventricular valves. After all, that's what the question is about. So the semilunar valves sit in the base of the arteries, obviously because we're told it's the left side of the heart, we're only interested in this side. And then the atrioventricular valves here prevent backflow of blood into the left atrium. So it's always worth keeping this in mind so we can actually understand what's going on here. So At the start of the cardiac cycle here, we can see the volume of the ventricle increasing. So that means that that ventricle here is filling. So once it's full, to prevent backflow of blood into the left atrium, we need to close the atrioventricular valve here, which is what number 1 is. So immediately I can write 1 here. And then as that left ventricle fills, we'll need to then contract the wall of the left ventricle in order to force blood into the aorta. So obviously the semilunar valves need to open to accommodate that, which is what number 2 is. So we know that we're now emptying that left ventricle of blood. So once that left ventricle is empty, we want to close the semilunar valves. So that will be 3. And then it all starts again. We want to then start refilling that left ventricle, so we need to open that atrioventricular valve, which will be number 4. Use the diagram above to calculate the volume of blood pumped per minute by the left ventricle. This is quite a lot of work for 2 marks. So first of all, you need to work out how long it takes for 1 cardiac cycle. And the way you do that is just look at your cardiac cycle here, and you just need to pick 2 points on the graph which are matching. Try and pick somewhere where it's easy for you to read off the x-axis. It's up to you, but I'm going to go here. So here are 2 matching places on the cardiac cycle. Then just read off, see how long that takes. So just work it out. There's 0.96 here, 0.2 here. To get the time of 1 cardiac cycle as being 0.76 seconds. Because we need the volume of blood pumped per minute, we need to find out how many heartbeats took place. So we do 60 seconds divided by 0.76, which is the time for 1 heartbeat, 1 cardiac cycle. So we know that 78.9 beats took place per minute. And then what is the volume per beat? We just need to work out the difference in volume between the maximum and minimum point. So that's between 120 and 40, which gives us 80 centimetres cubed. And so lastly, to calculate the volume of blood pumped per minute by the left ventricle, we just need to do the fact that we worked out 78.9 beats multiplied by 80 centimetres cubed equals 6,316 centimetres cubed of blood per minute. Explain the role of the heart in the formation of tissue fluids. So the heart, as I said, is going to be responsible for the high hydrostatic pressure at the arterial end of the capillary. And then we're going to say that this forces water, oxygen, glucose out of the blood capillaries. Forming tissue fluid, which bathes the cells. Lymphedema is a swelling in the legs which may be caused by blockage in the lymphatic system. Suggest how blockage in the lymphatic system could cause lymphedema. So remember, 10% of tissue fluid goes to form the lymph. That lymph eventually returns to the blood in the subclavian veins. So clearly a blockage in the lymphatic system would mean that excess tissue fluid would build up and not be returned to the blood. Give the pathway a red blood cell takes when travelling in the human circulatory system from the kidney to the lungs. Do not include descriptions of pressure changes in the heart or the role of heart valves in your answer. So just a simple description of the anatomy really of that circulatory system. So red blood cell travels. Remember, the word pertaining to kidney is renal. If it's leaving the kidney, it must be a vein. So that red blood cell travels in the renal vein to the main vein, which is the vena cava. At this point, it's going to enter the heart. It's going to enter the right side of the heart, the deoxygenated side. So it flows into the right atrium and right ventricle. Before leaving the heart. Remember, if it's leaving the heart, it must be entering an artery. Because it's going back to the lungs, the word meaning lungs is pulmonary. So it's going to leave the heart in the pulmonary artery. The figure below shows a section through two types of blood vessels observed using an optical microscope. I can see just from the shape this must be an artery, this must be a vein. Identify the type of blood vessel labelled M in the figure above. It's a vein. And why is that? We can see it has a wider lumen and thinner walls. Just have a look. There's the super wide lumen compared with the artery's narrow lumen, and the walls are much thinner compared with the artery. Tissue fluid is formed from blood at the arterial end of the capillary bed. Explain how water from tissue fluid is returned to the circulatory system. So this is everything I was talking about to do with the difference between the hydrostatic pressure at the arterial end versus the oncotic pressure at the venial end, which is responsible for returning tissue fluid to the blood by osmosis. We just need to talk about the latter part of that explanation because we're talking about tissue fluid being returned to the circulatory system. So how is it that the water potential is greater in the tissue fluid compared within the capillaries in the circulatory system? Well, it's all to do with those proteins which, remember, stayed in the blood. They were too large to leave the blood through the gaps in the epithelial cells. So we're going to say that plasma proteins remain in the blood, lowering the water potential at the venial end of the capillary. So water will leave the tissue fluid by osmosis. Remember though that this accounts for 90% of the water. 10%, remember, entered the lymph, and at the latter end of the lymph system that water is then returned to the blood. So don't forget to add that point. 10% of water re-enters the circulatory system via the lymph system. Figure 1 shows part of the blood circulation in a mammal. Use figure 1 to give the letter that represents each of these blood vessels. I think the most straightforward thing to do here is to start by labelling the chambers of the heart. Remember, the left and right sides are reversed. So that's the left atrium, left ventricle, right atrium, right ventricle. The blood vessels carrying blood away from the heart are always arteries. The left side of the heart is oxygenated. That means vessel D is the aorta. Whereas coming up here, you're going to have the pulmonary artery taking deoxygenated blood to the lungs. The pulmonary vein supplies oxygenated blood to the heart, and the vena cava over here takes deoxygenated blood to the heart. Anything to do with the kidneys is renal. This is coming off the aorta, so it's the renal artery. Taking deoxygenated blood away from the kidneys is the renal vein. That makes this bit of the question nice and straightforward then. What is the aorta? I've already labelled it as D. The renal vein I can see as G. The vena cava is F. Name the blood vessels that carry blood to the heart muscle. So supplying the heart with its own oxygen requirements are the coronary arteries. Moving on to mass transport in plants. So first of all, why do plants require a transport system? So the reason remains similar to why animals such as ourselves require transport systems, and that's because their surface area to volume ratio is too low, and therefore diffusion occurs too slowly in order to deliver the various substances the plant needs. So make sure you simply point out that the stem, roots, trunk have too small a surface area to volume ratio and diffusion is too slow. Secondly, what is the purpose of the transport systems? Clearly to deliver substances including sucrose and oxygen, which is moved from the leaves to the roots, mineral ions which are absorbed at the root hair cells upwards to the leaves, and then lastly make sure you just point out that plants can be very large indeed, and therefore obviously they require a transport system. After all, that will move all the necessary substances. What is a dicotyledonous plant? It's a plant that has two cotyledons in its seed. Cotyledons are organs which store food for a developing plant. The vascular system is simply the name given to the transport systems found in the root, stem and leaves, and the vascular system includes the xylem and the phloem. The vascular bundle is the arrangement of the xylem and phloem tissue. Let's take a look at the vascular bundles found within the plant at various organs. So we're taking the stem. Now, as you can see, in terms of the arrangement, you'll find that the xylem sits on the inside versus the phloem, which sits on the outside. Remember, the phloem transports sucrose and amino acids, while the xylem is transporting water and mineral ions. So collectively, these two tissues are known as a vascular bundle. Between them sits the cambium, and then on the edge of the stem you have the epidermis as well as the cortex. And whilst we have the diagram here, over here you see a light microscope that's been stained in order to view these various tissues. Here's your xylem, your phloem, your epidermis, and then your cortex sits here. As we move down to the root, you have the xylem and phloem arranged slightly differently. The xylem, it looks like an X. So try and use that to help you remember so that you can identify it more easily in the exam. At the xylem, you've also got the phloem. There's your epidermis as well as your endodermis. Your cortex remains very similar. And this is representing your root, which has a large surface area to absorb mineral ions and water. And then just pay attention here to the endodermis and Casparian strip, which I'll talk about very shortly. Again, here's a light microscope. You've got your xylem and then your phloem. And then last up, your leaf. Let's make sure you're happy with the view we're looking at. We're just going to draw a leaf here. The thing you'll notice about most leaves is the veins. So you're looking at the side view of the leaf and kind of looking at this bit here, the main vein. That's what the midrib here is representing. And you'll have your vascular bundle again with your xylem and your phloem, and then towards the top of the leaf you have your palisade mesophyll, which remember contains lots of chloroplasts that trap light for photosynthesis. Now, the purpose of the xylem is to transport water and mineral ions from the roots upwards, up the stem, to the leaves. So transport occurs in one direction only, that's upwards. As well as allowing the delivery of water and mineral ions, the xylem helps provide mechanical support, so helps to keep that plant upright. After all, it has no bones. How does the xylem structure link to its function? First of all, the xylem is made up of a continuous column of dead cells. The xylem contains lignin, which is a tough material that helps to strengthen the xylem, and this lignin can be arranged in either spiral or annular ring patterns. And the last thing to note is that the xylem have pores or pits in their outer cellulose cell wall, and that enables water and mineral ions to leave so that it can move into adjacent leaf cells or into other xylem tissue. Diving into a little bit more detail to do with the lignin, which remember is a tough substance used to strengthen the xylem. It helps to support the xylem and prevent its collapse, which may be likely to occur due to the tension produced by the transpiration stream found within the xylem. Remember, transpiration is the loss of water at the leaf surface via the stomata. Because the water molecules are all attracted to each other by hydrogen bonds, it forms a continuous column within that xylem. So as water leaves the leaf by transpiration, more water is drawn up from below, and that's creating tension within the xylem, meaning that the xylem needs reinforcing to stop it collapsing. The lignin also helps to waterproof the cell, which makes sense, after all the xylem is transporting water, and the lignin also helps provide adhesion between the water molecules and the wall, and that's important for the reasons I just said to do with transpiration stream. Those water molecules do need to be attracted to each other as well as the wall of the xylem. Take a look at the structure of the xylem. Remember, the lignin structures can either be spiral or in rings, the annular pattern. If we look at this diagram over here, remember the xylem is made up of a continuous column of dead cells. There are pits or pores in the outer cellulose cell wall, which allows that water to leave the xylem and flow into adjacent leaf cells or into xylem vessels. The water molecules are attached to each other due to the hydrogen bonds that occur between the highly electronegative oxygen and hydrogen element. Remember, this is kind of a more chemistry point. Those hydrogen bonds also help with adhesion to the side of that xylem vessel. And we're pointing out here with the arrow that water and mineral ions will always be moving upwards in the xylem. But why is that water so important? Why is the plant going to so much trouble to transport water around itself? Firstly, because that water provides turgor, hydrostatic pressure. Remember, I said plants don't have skeletons, so they need this pressure in order to stay upright. It helps support the stem, and it provides force for the roots to push through the ground in order to anchor the plant and obtain mineral ions. The loss of water by transpiration actually helps to cool the plant, Water acts as a solution to help move mineral ions through the plant from where they're absorbed to where they're used. And lastly, remember, water is a reactant in photosynthesis. Water plus carbon dioxide produces glucose and oxygen. So as well as water, the xylem transports mineral ions. Now the mineral ions enter the plant at the root, specifically the root hair cell. Because those ions are moving from an area of low concentration to an area of high concentration, this movement occurs by active transport and requires ATP, as well as protein pumps. The mineral ions which are absorbed include nitrates used to build plant protein, magnesium ions used to make chlorophyll, phosphates used to make ATP and DNA. Remember, water enters the root hair by osmosis from an area of high water potential, that will be in the soil, to an area of lower water potential found within the root. The lower water potential in the root is due to dissolved mineral ions, sugars and amino acids. So that's causing the lowering of the water potential in the root compared with the surrounding soil. Remember, this occurs across a partially permeable membrane. But how does that water move across the root? Well, there's several different pathways. The first pathway is the symplast pathway, otherwise known as the cytoplasmic pathway. The second route is the apoplast pathway, otherwise known as the cell wall pathway. Plasmodesmata refers to continuous cytoplasm channels that link plant cells, and they'll have an important role to play in the movement of water. So we'll dive into the symplast cytoplasmic pathway first of all. So water moves through the cytoplasm by osmosis. The plasmodesmata link adjacent cells, and because that water is moving by osmosis, we need to maintain a water potential gradient, causing that water to move from an area of high water potential to an area of lower water potential. And that water potential gradient is maintained by water leaving the roots and entering the xylem. The apoplast cell wall pathway now, water moves through the cell wall and into cellular spaces. The gaps between the cellulose fibres are filled with water. Remember, because of the hydrogen bonding, the water molecules have very strong cohesive forces, and these cohesive forces help to pull the water along, creating tension and therefore continuous flow of water through the cell walls of the plant. Touching on the Casparian strip. Now, the role of the Casparian strip is to control the movement of water, and it prevents water moving through the apoplast pathway. So it prevents water moving through the cell wall pathway and forces water to move through the symplast pathway, the cytoplasmic pathway. And how does it do that? Because the Casparian strip contains a layer of suberin. This is a very waxy material which lines cells in the endodermis, providing a waterproof layer around the xylem. So let's pull together all these things I've just discussed. So how does water enter the xylem? Water which was originally in the apoplast pathway, so the cell wall pathway, is forced into the symplast pathway, the cytoplasmic pathway, by the Casparian strip. Obviously, in order to do that, the water must pass through the cell membrane because it's moving from the cell wall to the cytoplasm. Remember, those cell membranes are selectively permeable, and that's important because it prevents toxic solutes entering the cells. Mineral ions are then actively moved into the xylem. Remember, the xylem transports both water and mineral ions. And then because of the presence of those ions, it lowers the water potential within the xylem, meaning that water enters from the cytoplasm of the cells into the xylem down the water potential gradient by osmosis. And this is known as root pressure. Take a look at this diagram now showing the movement of water through the roots. If water moves by the cell wall route, remember that's the apoplast pathway, the symplast pathway ensures water passes through the cytoplasm, and it's these plasmodesmata here, which are cytoplasmic channels that allow the continuous flow of water. A deep dive into transpiration. So transpiration is the loss of water vapour from the leaf via the stomata. And we can quite easily measure the rate of transpiration in a leafy shoot using a device known as a potometer. So if you have a look at this diagram of the potometer, you can see the leafy shoot is placed into a capillary tube. And what's going to happen is that reservoir is going to supply the leafy shoot with water. The leafy shoot absorbs water, and then as transpiration occurs through the leaf of the shoot, more water will be drawn along, moving that air bubble from right to left. And we can measure the rate of transpiration over a set time period. But just one thing to notice is we're assuming that the rate of uptake in that leafy shoot is the same as transpiration. There will be small water losses as that plant uses that water for photosynthesis. But for all intents and purposes, we assume that the rate of water uptake and the rate of transpiration are the same. A couple of things in terms of setting up the apparatus. It's important that we first of all cut the stem at an angle, and that's to avoid introducing air bubbles into the xylem. We need to make sure that if the leaves get splashed, that they get dried, as that will affect the rate of transpiration. Anywhere where there are joints, we need to seal those with petroleum jelly, again to stop the introduction of air bubbles. And obviously, as always, in terms of improving reliability, we need to repeat our experiments several times. Transpiration stream, this is the movement of water from the root to the leaves. It occurs in the xylem. The role of the transpiration stream is to help cool the plant, to deliver water and mineral ions to the leaves, as well as providing support to the plant. Now, the physical properties of water make it possible for transpiration stream to occur, and that's due to the hydrogen bonds which hold the water molecules together in a continuous column within the xylem of the plant. The water molecules are cohesive to other molecules as well as the inside of the xylem walls. And there's adhesion between the inside of the xylem walls and the water. So as water loss occurs at the stomata of the leaf, more water is drawn up from below due to the cohesive properties of water. Tension is created when water evaporates out of the stomata, and that's in part due to lower pressure at the top of the xylem. So obviously, I briefly described how water moves up the stem of a plant, but now in high-level biology, we need to describe that movement of water using the cohesion tension theory. So we know that water will enter the xylem down its water potential gradient. As the water enters the xylem, root pressure at the bottom of the xylem helps to push the water up. Remember that root pressure is brought about by the active movement of mineral ions into the xylem, which causes a lowering of water potential. If you think about the top of the xylem now, transpiration that occurs at the leaves creates low pressure at the top of the xylem, so you have a difference in pressure at the top and the bottom of the xylem, therefore creating tension. As we keep saying, the water molecules are attracted to each other due to hydrogen bonds, and that continuous column of water is pulled up the xylem by this tension. An adhesion of water molecules to the walls of the xylem helps support the column of water further. But what is the evidence supporting the cohesion tension theory? The most obvious piece of evidence is if there's a break in that xylem wall and air bubbles are introduced, the plant can no longer draw water up its stem, and that's because the continuous column of water is broken. A second piece of evidence supporting the cohesion tension theory is the fact that in trees, their trunk diameter is smaller in the day. And the reason for that is, remember that during the day when it's sunny, the stomata open in order to introduce more carbon dioxide into the leaf for photosynthesis. As a result, more transpiration, so more loss of water occurs, and therefore tension within the xylem is at its highest. And it's that tension which pulls the stem or the trunk of the tree inwards, creating a smaller diameter. So yes, the fact that the trunk diameter is smaller in the day provides evidence for the cohesion tension theory. How does the water travel from the leaves to the stomata? Well, it's by the symplast and apoplast pathway, which we met when we looked at how water crosses the roots. The symplast pathway involves the water crossing the cytoplasm via the plasmodesmata. The apoplast pathway involves water movement via the cell walls. The opening and closure of the stomata is controlled by guard cells which surround the stomatal pores, and they can close the stomata if excess water loss is occurring, which may lead to the plant wilting. So how does this opening and closure of stomata actually occur? Well, when conditions are favourable, solutes are actively pumped into the guard cells, lowering the water potential, meaning that water is drawn into the guard cells by osmosis. This increase in turgor of the guard cells causes the opening of the stomata, meaning more transpiration can occur. As we've said, the plant can choose to open and close its stomata based on environmental conditions. However, it is impossible to totally avoid transpiration during the day for the reasons I've already mentioned. The fact that the stomata open in order to allow CO2 into the leaf for photosynthesis means that transpiration will occur. And on hotter days, just due to the fact that water molecules have more kinetic energy, you'll find that evaporation occurs more quickly, meaning more transpiration takes place. What factors affect the rate of transpiration? Well, that's going to be humidity, wind speed, temperature, and light intensity. So if we look at increased humidity levels, why does that decrease the rate of transpiration? Well, there's more water vapour in the air surrounding the leaf. This decreases the concentration gradient of water inside the leaf and the surrounding air, meaning that evaporation occurs more slowly. On hotter days, transpiration increases because the water molecules have more kinetic energy, so there's a faster evaporation rate. On days where the light intensity is very high, very sunny days, more stomata will open in order to maximise the amount of CO2 that can be absorbed for photosynthesis. After all, sunlight is needed for photosynthesis, therefore meaning that water will automatically evaporate out of the stomata. And obviously it makes sense that during the nighttime, transpiration rates slow, After all, the stomata can afford to be closed as no photosynthesis will be taking place. And then lastly, increased wind speeds will increase the rate of transpiration because the water molecules are blown away from the surface of the leaf, increasing the concentration gradient, meaning that transpiration occurs more quickly. We've talked a lot about the structure of xylem, but what about phloem? First of all, remember the role of phloem is to transport sugars, so sucrose, and amino acids from where they're made, that's in the leaf, the source, to where they're used or stored, known as the sink. The phloem is made up of a combination of sieve tube elements and companion cells. Plasmodesmata, which are cytoplasmic extensions, link the sieve tube elements and the companion cells, allowing the movement of metabolites. Specifically, the sieve tube elements are long and narrow cells which are joined together to form the sieve tubes. They're connected by porous sieve plates at the end of the sieve elements. Although they're not hollow in the way that xylem are, they have reduced number of organelles and no nuclei. This is to maximise the space available for the translocation of materials. They have little cytoplasm and they have thick and rigid cell walls. The second component of the phloem is the companion cells. I really like the name of these cells. They're basically like friend cells, which actually help the sieve tube elements move sucrose and amino acids. And the way they do that is by having a large number of mitochondria. This provides the ATP needed to actively transport substances between the sieve tube and the source and the sink. The infolding of the plasma membrane provides a larger surface area to volume ratio to allow for more material exchange. Transport proteins located within the plasma membrane move materials into and out of the sieve tube, and plasmodesmata between the companion cells and the sieve tubes allow the movement of substances due to the extension of the cytoplasm. Take a look at the diagram of the phloem. So the phloem, as we said, is made up of a combination of sieve tube elements, as seen here with the sieve plates, and then they're surrounded by the companion cells, which we know contain an awful lot of mitochondria. The plasmodesmata, remember, are cytoplasmic processes which allow the movement of substances. Adding more detail now to our definitions of sources and sinks. So the source, this is the site where the loading of amino acids and sugars into the sieve tube occurs. And this is obviously going to be where these organic substances are made, where they're synthesized, so the leaves. The sink is where these assimilates are unloaded for use or for storage, for example, roots, fruits and seeds. In the next part of this video, I'm going to teach you how sucrose is loaded into the phloem, how water is loaded into the phloem, because the sucrose needs to be dissolved in water for it to move. And then we're going to combine that just to talk about the overall story as to how the organic compounds are transported. So how is the sucrose loaded into the phloem sieve tube elements? Well, we need a lot of information here. So you want to start by saying that protons, which remember are H plus ions, are pumped out of the companion cells into surrounding tissue. This is an active process requiring ATP. And the movement of these protons creates a proton gradient. What happens next is, because of the H plus gradient produced, the protons, the H plus, are able to re-enter the companion cells, this time by diffusion, and they're going to be attached to the sucrose molecules as they move into the companion cells. So the protons and sucrose both flow through co-transporter proteins in the plasma membrane, so we say that they move by facilitated diffusion. So far the sucrose has entered the companion cells, but how does the sucrose enter the sieve tube elements? Well, it's by diffusion and it's via the plasmodesmata. And do note that amino acids can also be loaded in this way. So we've talked about how sucrose loads into the phloem at the source. How are we going to add water to the phloem? Well, that high concentration of sucrose that was loaded into the phloem sieve tube elements at the source obviously lowers the water potential at that point in the phloem. Remember that the xylem and the phloem are running parallel to each other. So what happens then is that water moves across from the xylem into the phloem, down the water potential gradient by osmosis. And that has a knock-on effect of increasing the hydrostatic pressure at the top of the phloem, so near the source. And then obviously because the sucrose hasn't moved yet, comparatively speaking, there'll be a low concentration of sucrose at the sink. So for example, the roots. So what does that do? So what that does is that it increases the water potential at the sink. So you've guessed it, water this time is going to leave the phloem and enter the xylem by osmosis. And that's going to lead to a decrease in hydrostatic pressure. And then finally make a point, which is that water moves down the hydrostatic pressure gradient. And the reason for that is due to a physical property of water, which is its incompressibility. So here's a nice summary of how water moves in the phloem. So here in my revision guide, we can see a combination of how sucrose is loaded into the phloem, how the water enters, and how fundamentally that sucrose will therefore move from the source to the sink. So we're saying here that phloem transports organic compounds such as amino acids and sucrose. As we know, they're going to move from the source, which is where they're made, to the sink where they're stored or used. This is known as translocation. So it occurs in the sieve tubes of the phloem. We know that hydrogen ions, protons, are going to be actively transported, so that's going to require ATP, which is produced by the many mitochondria in the companion cells of the phloem. By creating this proton gradient, what happens next is that the protons will diffuse back into the companion cells, but this time they're going to bring sucrose with them, and that's going to occur by facilitated diffusion using co-transporter proteins. And then as a result of that increased sucrose concentration at the source, that's going to lower the water potential, meaning that water is going to enter the sieve tube by osmosis, causing a high hydrostatic pressure at the source. And then remember, at the sink, we have a comparatively lower concentration of solutes, of sucrose, meaning that you have an increased water potential at the sink. So water leaves the sieve tube of the phloem and moves into the xylem by osmosis, and we call this translocation or mass flow. And then make sure in the exam if they ask you about glucose, so it says here, outline how glucose produced in photosynthesis is transported and stored in plants. Really, it's a question to do with how that glucose, which is turned into sucrose, is moved in the phloem by the process of translocation. So again, it's kind of reiterating everything we've already said. We know that it's an active process whereby sucrose is loaded into the phloem. We know that the sucrose moves from source to sink in the process of translocation. That source is going to be your leaves where photosynthesis takes place. Your sink could be fruits, seeds, roots, storage organs. And then when that sucrose is going to be stored, it needs to be converted into insoluble starch so that it has no osmotic effect. If they ask you about how we can measure the rate at which transport of organic materials takes place in the phloem, you can use these insects known as aphids. You can see its sharp needle-like mouth parts known as stylets. They're going to pierce the stem of the plant and they're actually going to poke into the phloem so that the aphid can steal all that sucrose in there, which it can use for its own growth. So scientists can manipulate this to actually examine phloem transport rates by its supermean. So what they're going to do is they're going to cut the aphid away from its mouthparts, and by doing that they're going to have inserted a tiny needle into the stem, which will then start oozing sucrose, and that's due to the high pressure inside the sieve tube pushing the phloem sap into that mouth part, and then they can obviously capture the sucrose and measure how quickly it's moving through the stem. And then because this part of the specification is super dull, I'm just going to leave a screenshot of my revision guide so you guys can scribble down the notes to do with more evidence supporting this mass flow theory within the phloem. A scientist measured the pressure in a phloem tube in a willow plant stem. He repeated his measurements to obtain 9 readings. His results are shown in the table below. The percentage error of the mean phloem pressure in this phloem tube is calculated using this equation. Percentage error equals uncertainty in measurement divided by mean times by 100. The uncertainty in measurement is half the range of the measured values. Calculate the percentage error of the mean phloem pressure in this phloem tube. Show your working. So they've provided us with the equation. Let's start by finding the uncertainty in the measurement, which we've been told is half the range of the measured values. So to find the range, we need to take the highest value and then take away the lowest value. So look along, it's going to be 7.0. So that means the range is 9.3 minus 7.0 equals 2.3. And then it says that it's half the range, so we need to divide that number by 2. So we're nearly there. Percentage error equals uncertainty in measurement, which was 1.15, divided by the mean. So to find the mean, we need to add up all these values and divide them by 9. So the total is 73.8. Divide that by 9 to get a mean of 8.2. Multiply by 100, and your final answer here is 14%. The mass flow hypothesis is used to explain the movement of substances through the phloem. Use your understanding of the mass flow hypothesis to explain how pressure is generated inside this phloem tube. That's worth 3 marks. So that pressure generation is going to be due to water moving into the phloem. So first of all, we need to write how that comes about. So remember, the first thing is to talk about the fact that the sucrose is actively transported into the sieve elements of the phloem by the source. This increase in solute concentration, remember, lowers the water potential, meaning that water is going to enter the phloem from the xylem by osmosis. The scientist also measured changes in the phloem pressure and changes in the rate of water movement in the xylem of a willow plant at intervals during the day. His results are shown in the graph below. So we have phloem pressure, here's the time in hours, and then we have the rate of water movement in the xylem. You can see that it's at its maximum when it's going to be the hottest, sunniest portion of the day, most likely. Describe the relationship between phloem pressure and the rate of water movement in xylem in this plant. So because it's a describe question, we need to say what we see in this graph. So you can see that the phloem pressure reduces as the rate of water movement in the xylem increases. Phloem pressure is reduced during the hottest part of the day. Use information in the graph above along with your understanding of transpiration and mass flow to explain why. So remember, the phloem and the xylem are running parallel to each other. Because of the pores in the walls of the xylem, it means that water can enter and leave the phloem. So let's work out what's happening in terms of transpiration at various points of the day and how that will link with mass flow in the phloem. So we're going to start by saying, in the hottest part of the day, the highest rate of transpiration out of the stomata occurs. Remember, this means that the tension within the xylem will be at its highest. And then you can infer here, when it says the flow and pressure is reduced during the hottest part of the day, we can infer that less mass flow is taking place in the phloem, and that must be because there's less water moving from the xylem into the phloem, creating that hydrostatic pressure. Describe the cohesion tension theory of water transport in the xylem. So we're going to write an answer here which combines transpiration stream as well as a little bit of chemistry to do with how water molecules are attracted to each other by hydrogen bonds, forming a continuous chain within the xylem. So we need to start by first of all pointing out that water is lost at the surface of the leaf via the stomata by the process of transpiration. Water molecules are attracted to each other due to hydrogen bonds. There's a continuous column of water within the xylem. Very similar point, just point out that there's the adhesion of water molecules to the walls of the xylem. And then due to transpiration stream, water is pulled up the xylem. A student used a potometer to measure the movement of water through the shoot of a plant. The potometer is shown in figure 1. As water is lost from the shoot, it is replaced by water from the capillary tube. In one experiment, the air bubble moved 7.5 millimeters in 15 minutes. The diameter of the capillary tube was 1 millimeters. Calculate the rate of water uptake by the shoot in this experiment. Give your answer in millimeters cubed per hour. Show your working. The area of a circle is found using the formula area equals pi r squared. So we need to work out the volume of water moved per hour. So we need to start by finding the area. So if I draw a little diagram, if this is your water, in effect you need to find out the volume of water. So how do we do that? Well, we work out the area of this circle here, pi r squared, and then we multiply it by the length of that capillary tube, which we were told was 7.5 millimeters. So to find the volume, we're doing pi r squared. We were told that the diameter of the capillary tube was 1 millimeter, so the radius is therefore 0.5. So we do pi times the radius squared, multiplied by the length of the capillary tube, which was 7.5. And then we need it per hour, so just multiply 15 minutes by 4. So to two significant figures, our answer is 24. The student wanted to determine the rate of water loss per millimetre squared of surface area of the leaves of the shoot in figure 1. Outline a method she could have used to find this rate. You should assume that all water loss from the shoot is from the leaves. So if we have our leaf here, first of all, how do we work out the area? Well, just place your leaf on graph paper and count the squares. Obviously, there are two sides of the leaf, so make sure you do double it. And then lastly, to find the rate of water loss, you need to take water loss and then divide it by the total surface area of the leaves. The rate of water movement through a shoot in a potometer may not be the same as the rate of water movement through the shoot of a whole plant. Suggest one reason why. Well, what's different between the setup of a potometer and the shoot in a whole plant? Well, look, we don't have any roots here. Describe the mass flow hypothesis for the mechanism of translocation in plants. So translocation is all to do with the movement of sucrose in the phloem from source, so where that sugar is made, to where it's either used or stored, which are known as sinks. So we just need to add a little bit of extra detail as to how those substances are moved. So translocation is when sucrose in the leaves, so there's our source of sucrose, and it does require energy from ATP, so we're going to say is actively transported into the phloem. Remember, that's due to the companion cells on the side of those sieve tube elements, which have lots of mitochondria to provide the ATP. Because of the movement of sucrose, what that does is it lowers the water potential of the sieve tube element of the phloem. Meaning that water enters the phloem by osmosis. And due to that water entering, what that does is it increases pressure within the phloem, therefore causing mass movement of the sucrose towards the sink, which could be the roots, which take that sucrose and convert it to starch for an insoluble storage solution. Scientists measured translocation in the phloem of trees. They used carbon dioxide labelled with radioactive 14 carbon. They put a large clear plastic bag over the leaves and branches of each tree and added 14 CO2. The main trunk of the tree was not in a plastic bag. At regular intervals after adding the 14 carbon dioxide to the bag, the scientists measured the amount of 14 CO2 released from the top and bottom of the main trunk of the tree. On the surface of the trunk of these trees, there are pores for gas exchange. The following figure shows the scientists' results. So here we have the mean amount of radioactive carbon in the carbon dioxide released from the trunk of the tree. This graph line refers to the top of the trunk. Here's the bottom of the trunk, and this is the time after the 14 carbon labelled CO2 was given. Name the process that produced the 14 CO2 released from the trunk. Finally, a nice straightforward question. Where does that CO2 come from? Remember, it's the tree respiring. It is a living organism after all. How long did it take for the 14 carbon label to get from the top of the trunk to the bottom of the trunk? Explain how you reached your answer. So you need to look for two matching portions on the graph. So we'll take these two peaks here, just work out the difference between them using this x-axis here. It is a bit of an awkward axis, but the answer here is about 30 hours. And we're explaining our answer by stating it's the time between peak of 14 carbon at the top of the trunk and the bottom of the trunk. What other information is required in order to calculate the mean rate of movement of the 14 carbon down the trunk? Clearly, we need to know the length of the trunk.